All right, so sorry about that. We lost a few minutes at the beginning, forgetting to hit record. But this is day 3 of the workshop 6 for the UCLA QC Biochemistry. That's the BS-Seq analysis for February of 2021. Okay, so let me start the screen sharing up here. Okay, we need keynotes, we need paraterminals, we need IGV. We'll need IGV later, maybe we'll save that for later. Okay. All right, so we're slowly building up here to our usual situation. Chat window floating over here somewhere. Got terminal window going. Okay, let me start that fresh. Then does this, I should restart screen sharing just to make sure that guy gets included. So, all right, so start up your Hoffman if you haven't already. All right, and change to my scratch space. And there's a copy of day 2 now that's got sort of everything settled from what happened, you know, just sort of cleaning up from what we did there. Oh, and don't forget, there's going to be a quiz and a homework for those of you taking it for credit. And I will email those to you tomorrow. And the quiz is due by the end of day Monday, and the homework is due by end of day Tuesday. And the way you get things back to me is just email your responses back to me. My address is at focus@ucla.edu. Okay, so don't forget to do that if you're taking it for credit. And the evaluations that Eli talked about are appreciated too. Right, so we yesterday we got through kind of 2 rows of this pipeline overview chart, and we're now at this point where we're ready to start doing statistics and combine the data from our different samples together to start examining conditions rather than samples. We have 2 replicates, 3 tissues, and then at the end we want to get to some DMRs and stuff like that. So we need to, okay, so here we are, we're on Hoffman. Today you need less resources than yesterday. You can get away with 2 cores and 5 gigs of RAM per core, so maybe 10 gigs of RAM. You probably don't even need this much, but if you're waiting for resources, if you ask for more than this and you already got a session, that's no problem. But if you if you're waiting a long time or whatever, you can reduce the cores to 2 and the RAM to 5, 10 gig total, and hopefully that gets you faster. Today the resource demands are quite a bit lower than yesterday. Oops. There we go. Make sure we're on a compute node. We don't have to wait long here. There we go. Oops. Okay. So, you know, again, just our usual pattern each morning here. That's your personal scratch space. Right? Here's changing into my scratch space, the workshop subdirectory. And there's now a day 3 start state. You copy preserving permissions, because some of those files have execute bits and so on, and you want those to live, you know, retain execute bits on your side. Recursive, progress information, if there's any conflicts, let us know. And copy my day 3 start state into your place as a new directory, day 3. Okay, so let that go. And while that goes, we'll do some talking. So the bugs in BSBall I discovered are, one of them is not a bug. Okay, one of them is just I'm not sure whether it's supposed to guarantee that the CGMAP files are sorted by chromosome name and coordinate. It seemed to be in our case. I think I've had it not do that in the past. I think it's related to sort of how the internal parallelization threads work. Safest thing to do is to just, in your work script for the generation of the CGMAP files, is we'll just explicitly sort it ourselves. And I've modified the script to do that now. So now we know it's sorted and we don't have to think about it. It's just basically a good rule in bioinformatics is that any assumption that you have not explicitly verified yourself has a likely possibility of being false. It's sort of just over time, it just sort of seems to be true. So yeah, and for assumptions that you're likely to make that may or may not be true, you might as well just do the work to ensure that's true and not have to worry about it or have your analysis behave in some strange way that you discover later, possibly months later, and you realize that you have to redo everything because some point in the middle was didn't meet your assumption, and you didn't realize it at the time. The next problem is a bug in BS-Bolt, and that's I don't know if you want to call it a bug in BS-Bolt or not. It's effectively a bug in BS-Bolt. But BS-Bolt uses this PySam Python package to deal with the input alignments at the column methylation stage. It's reading a BAM file, and there's this PySam library that it's using to do that, read up that BAM file, and in particular, there's a pilot command inside that thing that it uses. And there's an argument to that pilot command that tells the maximum depth of coverage that it should deal with. Doesn't seem to work. And so what happens is that the max coverage parameter, which I think we set to 999 or something, whatever, basically doesn't work in BS-Bolt. And so if you look at the CGMap files that we got out, there are sites in there whose coverage is over 999, which doesn't make sense given the command line parameters we asked for. So all I did also to our CGMap generation script is that I just post-filtered the output of BS-Bolt just to enforce that. So if there is a site that comes out with too high coverage, exceeding the parameter we gave, we just filter it out. So we effectively work around that bug. I'll let the guys know so that hopefully they can work around it internally. So it's very easy, right? They just have to check when we emit the line whether it's really going to pass or not. Okay, my copy's done here, and I'm going to switch now to the working copy, and this would now be in your personal place. So we're ready to go today. All right. So remember, these are the CGMap files. This is sort of the end stage of the primary analysis. And we've got a CG file, a CHG, and a CHH file. It's basically the chromosome name, which strand, because everything's quoted in the plus strand. So the C's reference genomic C's, which are plus strand guys, and the G's represent genomic C's on the minus strand. So effectively represents a minus strand entry. These are coordinates for which genomic cytosine we're talking about. These two columns now are less useful because we've broken up the files, but I don't want to change the column numbering, so we'll keep them as is. So that's the context there, and then a start of information about subcontext on the next column, and then the last three columns are the actual data, right? So that is the point estimate of fraction methylated in the next, in the, what is it, 1, 2, 3, 4, 5, 6 column, and the last two columns are the numerator and denominator for that thing as discrete counts, right? So 1 out of 1, that's at 1.0, 100%. 0 out of 2, that's 0%, and none of the examples at the beginning of the files have any interesting numbers, but we saw numbers in the middle yesterday. Okay, so that's a little reminder of where we're at. And the CGMap file, these map files aren't so convenient to use because mainly you're always going to be dealing with many samples in your analysis, and each sample's in its own file, and then, well, that'd be okay, except for the fact that the subset of lines that appears is only the places where the coverage in that particular sample meet our thresholds. And so there's no, the different files from different samples won't have their lines in sync, right? I mean, because it's like the subset of sites that are present is going to vary depending upon the sample. And so that's sort of inconvenient because when you do your analysis comparison and so on, obviously you need to know what the value for all the samples are or whether the past thresholds are at a particular site, right? You need it, and which could be all lines all over the place in the different files. So we sort of want to bring the files together into one file that just has, a single row per position in the genome, and then has all the data for all the samples associated with that, rather than having to look up randomly in all these files. Okay. Right. So that is the BS-Bolt aggregation module, which is convenient for us to use. So we got to, we're using BS-Bolt again. We started a new session today. We got to get Python already. We got to put the Python packages bin directory in our path. And now we can use BS-Bolt again. And the module is called aggregate matrix. Okay. So bang, that is the command line help for that. And it's pretty simple. You got to give it the list of CGMap files you want it to merge. you got to give it the sample names to use as the column labels for those different guys, because the file names usually are not very nice and so on. And right now we've only got the SRX accession numbers in there. We actually need to know for the analysis which one is the neural progenitors and which one is the mesenchyme and so on like this. So we need sample names that make sense and are reasonable to use. So we take the parameter for those. And then as we talked about on day one, when we do these analyses, we're now going to have to make a decision about what sites in the genome we're going to analyze. And that is, not every site in the genome will have every sample have coverage or have every sample have coverage in a range that we like. And so we have to pass to a subset of sites in the genome that are going to be reasonably complete and of high enough coverage in enough libraries that we're going to be happy with analyzing. So that the two thresholds that are in BS-Seq aggregate matrix are min coverage, which is a per sample thing. So we're not talking at a single cytosine, whether this is going to make it to the aggregated file. And the min coverage thing says for each sample, you got to have at least this much coverage. And if you don't have that much coverage, you're considered to be missing for this site. Okay. Now, it's not necessarily death to the site if one sample goes missing or something. That's what the next parameter is going to control. But this first parameter controls whether each individual sample is considered to be present or absent at this site. Okay. So first, maybe we'll set it something like 5 or something, right? We can't be too greedy, right? We know we have fairly low coverage data here. Maybe we'll set it to 4, whatever, something like that. But we can't set it to 10. I mean, that's probably, we don't have much data over 10 probably. But we haven't looked at the coverages either. We got to do that too. So the second parameter, min sample, is related to how many missing samples you're willing to tolerate. So if you set min sample to 1.0, I don't like this parameter for a couple of reasons. And this is actually where another bug in BS-Seq comes in. So it's not given as an integer number of samples, like kind of would make sense and be the easiest thing to specify. It's instead input as a fraction of the samples that's required to be present. Okay. So if you make that 1.0, that should mean you're requiring all samples to be present, and basically you're going to toss any cytosine where any of the samples don't make the min coverage cut, right? So you're going to have only a complete matrix that comes out at the expense of throwing away possibly lots of rows, right? If you make that 0.0, I guess, then you're saying even one sample's presence, keep it, right? Just you'll have a lot of missing data in that row, but that's what you asked for. And if you set it to something in the middle, then we have to talk about how this translates from a fraction to the threshold, which is where the bug comes in. If you had set the fraction in some way that made sense, that we've got 6 samples, and if you had set it to 5, then once you get through this fraction business, but if it kind of turns out that it means 5 or more samples, then yeah, you're willing to tolerate the loss of 1. Some rows will have complete, and some rows will have 1 missing, and the 1 missing will bounce around, but nobody will have 2 missing, right? That's not allowed by your request. Okay, so now we can talk about this. We'll do that in a second. Okay, talk about the translation from the fraction to the threshold and why it's problematic. So here's our little template for command line for this thing, right? So BS-Seq aggregate matrix. There's this count parameter, which we'll talk about in a second. And then there's this -f, and this is where you name the CGMAP files you want to merge. And programs vary in how they take lists of things, right? Some programs would take this as you just say -f, and then you just list one file space, another file space, another file, and each file will become a separate command line argument. And it sort of knows when it reaches the next dash sign for a new option, that's sort of the end of that list. BS-Seq doesn't do it that way. It assumes none of your files names have commas in them. And it kind of also assumes that you have at least 2 of them. And then you just put a single comma between all the entries, right? So you basically say path to first file, comma, path to second file, comma, blah, 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 path, comma, path to last file, right? So these are the CGMAP files that you want to merge. They can be gzipped, I think, no problem. Then the -s is the sample names in sync with that, right? So same structure, commas separate the entries. If there were n entries in the file names, there got to be n entries in the sample names. And it's just name for sample 1, comma, name for sample 2, comma, name for sample 3, comma, blah, blah, name for sample n. And these names can't be too crazy, right? Because they're going to turn into column names in the output file. And we're going to probably read that with R and stuff. And see, you don't want to like get too crazy with weird characters in those, or even white space probably, and so on. It's just going to make your life hard. So we'll keep those simple. The -O then names the output file. Unlike all the other modules of BS-Bolt, that is not a prefix. It's the actual literal file name to put the output into. And that is a tab-separated plain text file that comes out. Also unusual for BS-Bolt, it is not Gzipped. Okay, so it's really just a naked plain text tab-separated file with some columns we'll talk about in a second. Then comes the min coverage, min sample thresholds that we were talking about. And the -T is the number of CPU cores you want it to use. This verbose option gives you a progress bar. If you're going to run it interactively, like we're going to run it in our session, our intro session, it's nice to use -verbose because it gives you a little progress bar as it works. Okay, yeah. So okay, what about this count parameter? Well, you can either include it or not include it, and the output file you get depends on whether it's included or not. If you include the -count, then the output file has 2 columns for every sample. One for the numerator of the point estimate methylation at that site, and one for the denominator. It's exactly the last 2 columns of one of the appropriate input CG map file. Okay, that's what that is. So every sample generates 2 columns. Without count, then each sample only produces 1 column in the output, and that is the fraction, the point estimate fraction methylation at the site, which is the 6th column in the CG map files. Okay, so we'll run it twice, once with and once without, and we'll keep both versions of these files. It kind of would have made more sense to me that why not just put 3 columns and just have the same data in there. Or really, the methylation fraction column isn't that terribly useful because you most always need to know the integer numerator denominator, and it's so trivial to recompute the fraction that it's like, why bother having all this complication with this extra column. But such as it is, and part of it has to do with interoperating with software that already took files of this kind of format from before and so on. History is always a powerful, inertia is always a powerful motivator. Okay, so yeah, so I keep mentioning there's this bug in min sample, which takes a fraction of samples, minimum fraction of samples necessary to keep the site. If you have, let's pick some easy numbers. Okay, so suppose you had 4 samples. We have 6, but 6 is horrible in decimal. 1 6, 2 6, well, 2 6 is a third, but they're all horrible in decimal, right? So let's use 4 because with 4 it's kind of 0, 0.25, 0.5, 0.75, 1 point. Right? That's the kind of the natural breaks for a 4 sample guy in terms of fraction of guys you have to get the site. So you'd expect that, well, because in a way, the 1/4 is not a good example because you can easily type 0.25, 0.5, 0.75, 1.1, 0.0, and they're exact. Say with 6 samples like we have, if you have if you want to have a threshold of 4 of them, say, then that's 0.666666666 forever, right? When you write it in decimal, the computer doesn't work in decimal inside; it works in binary. And 1 6 isn't a nice binary fraction either. It's infinitely repeating either. But it also binary doesn't really jive too well with decimal either. So if you truncate the decimal fraction, it doesn't, it kind of still makes a horrible binary fraction. And if you take a binary fraction with short number of digits, it's not necessarily a nice decimal fraction. And we don't really know how many digits BS-Bolt's using inside. It's probably Python is based on C, the C programming language, which on all modern machines uses IEEE 754 floating point numbers. It's probably a double, which is 64 bits long with 52 bits of precision, which is roughly 17 decimal digits, somewhere between 15 and 17. Then there's the details of how decimal to binary conversion is done for the floating point and so on. And there's a problem in that if we type something like 0.666, 3 digits, that's going to be probably lower than what BS-Bolt considers to be 2/3. If we try to put all the digits there, there's no guarantee that actually we're going to hit the last binary decimal place that's kept in the floating point number exactly the same way BS-Bolt is going to compare it. And so it's just very unstable to try to hit this fraction exactly when our interface is to type in decimals for things that are non-terminating fractions in decimal. So it means that we shouldn't really try to aim this fraction right at a spot. It'd be better to move this fraction in between the spots so that, you know, at least the ambiguity of providing the input and the conversion into the internal floating point number isn't really an issue. So if going back now to the kind of the 4 sample example, you know, the natural breakpoints are 0, 0.25, 0.5, 0.75, and 1.0. But let's not type in a 0.5 or 0.75. If we wanted a threshold of 0.75, probably makes more sense to give a number like 0.6 or something, or, you know, something halfway between 0.5 and 0.75, because then the fuzziness of converting that to the internal thing doesn't move you, you know, across a boundary point, right? If you put in 0.6, 0.75 is certainly above 0.6, and 0.5 is certainly below 0.6, and it's not really ambiguous, right? So it feels safe. Well, so that's what you think. So basically, that's the plan, right? If you want to have at least I out of n samples present, then the fraction you should give is like I minus a half on the numerator divided by n, right? You know, so that you kind of back off to the left a little bit to give yourself a little room for the fuzz of the floating point business. The problem is if you actually do that, then you get a threshold that is empirically 1 too low. So BS-Bulk gives you more more things missing than you think you should have allowed by 1. And it's essentially because it's rounding the fraction you give it in downward instead of upward. And so this is basically a bug, because it doesn't, the way it operates doesn't make sense given the help language for this thing. And so in the code, if you dig it out of the code, the line is wrong. It's rounding down instead of up. So that'll produce a little mess. In the shell script that we're going to run for this thing, this whole blob, which is a big comment, is here to explain this one little place where I have to put a minus sign, a plus sign instead of a minus sign, and it's to work around that bug so that the number you really want to give it for the threshold, which is literally the number of samples you want to at least be present, actually works the way it's supposed to be. So in the end, we'll work around it and provide a much nicer parameter to give. But that's the explanation for why we had to do that.
Sean, just a quick question.
Yeah.
So with this bug, presumably then it would be a systematic error. So if you were processing all your samples, then all of them will have an underestimation because, as you say, there's the rounding down issue.
But it's never good when you think you asked for at least 5 samples present, and then your output files have some with only 4, right? The files don't match your expectations, and you're going to wonder what went wrong, and so on. It's just, anyway. So I will tell this to the BS-Bulk guys, and it's an easy fix. It's just a point inside the code that converts the command line parameter needs to round up instead of round down.
Okay.
And we'll take care of it. But so okay, so we need okay. So this is we got everything ready. We got to compose our command and make the script and run it and all this stuff. It's probably too much to ask to try to pack all the knowledge about the file names and the sample names into the command line directly. So we're going to make a little file that has the pairings of those. So if you look in the copy, you should all have, hopefully their copies are done now, right? So somebody want to, did anybody get this? You've got a local copy now of the current day stuff? Yeah, okay, good. Should have been long enough. So we got this stage 6 to 7 key file, right? This is a tab separated file. I mean, it's sort of hard to tell whether those are spaces right there or not, but it's actually a tab. In the GUI terminal, you can kind of tell because the highlight, if I select there, doesn't want to go until it only goes all at once. So you can't select the things in between. That's how you know it's a tab. The other way to check is to use TR or something and convert the tabs to something you can actually see, like what's a character, like an exclamation point or something, some character that's not used there, right? So now you can see that is a tab there, and there's no extra tabs at the ends or anything anyway. It's a clean 2-column tab separated file. The sample names we want to use are in the front, and I just put the tissue first with abbreviating the tissue and an underscore, and then replicate 1, replicate 2. Okay, and this is going to be a very nice way to have it when we get to doing Methylation. Then the second column is the file name base that we want to use for this. It's not the literal file name because we've got some flavors, like do you want the CG flavor or the CHD flavor and so on. So rather than make separate key files for each of those cases, I just made the column for the file name be the static part that's not going to vary for us. So that is the stage 6 stuff that came out of the bisulfite quant. That's the directory that has the CG map files. Now that we've sort of fixed the thresholding for only keeping sites whose coverage is less than or equal to what we asked for, that's the max fix version. Because there was a point yesterday when I had multiple versions of things I was trying to figure it out, and I just had to add a suffix to some names to make sure I didn't confuse which version was which. So I've left the old one here as max broken, and the new one is max fix. Okay, so if you compare those two, you'll find that there's a few lines different in this. So we deleted some sites where the coverage was like in excess of 999. Okay, and we'll see today that 909 is higher than it. We should probably use a smaller number than that. But we know that after we'll do some stuff in R. So that's the directory where the CG map files are. Then here are the names, and I stop right at the point where, here, just to remind you what those file names look like for real, right? So here's here's the actual files in that directory. And that once you get to max 999, what comes next? Well, that's whether you want the CG underscore, the CHG, or the CHH stuff, right? So it's which context, basically. So that's what I covered off. The script will put, the script we use to run this aggregate matrix BS Bolt module, we'll put that back on. Because we're going to do all 3 flavors, of course. Not that it's terribly, super relevant for here, but certainly, you may be doing Rabbitopsis or whatever. I know at least one of you is Jacobson. So, all right. So, yeah. Okay. So that's that file. That's this key file, which is going to be available to our script, and that's what we're going to use to compose. Remember, we've got these arguments to BS Bolt aggregate matrix, this -F with the comma separated file names, and this -S with the comma separated sample names. And basically, our script is going to read this file and construct this command line arguments that way. Okay, right. So the script is down here, and we're just going to run it interactively in our session. We're not going to do any job submission stuff today. We're going to just work directly in our interactive session, and it's fine for our problem size. All right, so we're just going to set some variables. Yeah, numcores 2, because that's what I asked for today. Min site coverage, how about 4? Okay, so for a sample to be considered present as a site, we'll ask for minimum coverage 4. And the minimum number of samples that we'll use to keep is 4. So we'll let 2 samples drop out. So what's going to happen is that, peeking ahead a little bit, one of those tissues doesn't have its coverage completely in the same place as the other tissues. And so often what this 2 missing guys is going to be 2 replicates of the same tissue. So in a way, you can kind of think of it as allow one tissue to be gone, but there's nothing in the that's going to force the 2 missing guys to be from the same tissue, right? It's going to move around. I didn't want to make it 6 and just go for a completely solid file where all lines are present, because I wanted to show you in R how you handle the missing data case. So that's why I went down before. This is the name of our key file, just so we don't, so that's, just kind of one place to change it here. And the number of samples, which is something we're going to embed into the output file name, we just do that by counting the lines with wc-l in that file and capturing the result of that, running that command in the shell variable num samples. So actually, if I, we can sort of pre-flight this by, there we go. I got an extra A there somehow. Where did that come from? Actually, my A key is not only dying, it now seems to actually be sticky. So now sometimes I just get piles of A's everywhere. So if I do that, and then I ask, how many samples do I have? I have 6. Okay. Right, so here's where I use num samples. This is going to be the base for the output file names. Because we're going to run this with that count version and the fraction methylation version, we'll need to build a couple file names for the output. So this is the base output file name. Okay, so once you substitute it in the 6, we're going to do this. And rather, I decided it was too, there's a point where you can't really pack all the data in the file names, right? So rather than actually list the sample names in this thing, I just called it plex 6. It's a multiplex of 6 things together. And, there's a point where you can't let the file names grow to this big. And your operating system has a limit at some point. On Unix, Mac, it's usually around 250. On Windows, it can be. On Windows, I think it's more like 250 inside the OS, but the GUI doesn't like stuff beyond like 30. Okay. Now here's this crazy business here where we've got min number of samples we'd like to keep, but we got to convert that to that fractional thing that BS-Bolt wants. And so here's a way to do it in shell, right? So I'll show you the result of that, right? We're asking for 4 out of 6, right? Min sample fract. Okay, so why does that look wrong? Because I didn't set min number of samples yet. Okay, I kind of skipped that step. I didn't copy. I'm pre-flighting this by sort of playing with it interactively in the shell, but I didn't set all my variables yet. Okay, so I went back there, and okay, let's check out this line. This is sort of another way these scripts don't usually come out whole like all the first go, right? You sort of develop them interactively here, and then you're sort of working on the final version here. It takes a couple of iterations sometimes. So this is more of the kind of the working, while you're working out how the script's going to go, you kind of do it like this, and then as you get your lines right, you copy them down into your script file. Okay, so there we go. So 0.5833. Okay, so now I'm going to get out my calculator, which is going to be in a window that's not shared, and I'm just going to type 0.58 and a bunch of 3's, and multiply that back by our number of samples, which is 6. And it's like 3.5 out of 6, right? Which is exactly what I kind of want, right? Because if I say at least 3.5 out of 6 samples have to be present, 3 is a no, and 4 is a yes. And we have 1 and 4. Sounds exactly right. So the problem is if you actually do this in BS-Bolt, it allows 3 or more and not 4 or more. And it's due to the fact that it's essentially rounding its threshold in the wrong direction. So the way to solve it for us is to simply change this minus 0.5 to a plus 0.5, and that will work around the bug. But based on the documentation for what the interpretation of that parameter is, that's not what you're supposed to have to do. But okay, the fix is going to be simple. The little plus sign here. Describing the fix is going to take all these words, right? So that's the comment that kind of says all that, but the fix is really a 1-character workaround, right? So that minus becomes a plus. Okay, so we're going to do every context. So shell loop here for CTX is going to be CG_CHG, CHH. Those are part of the file names of our CG map files. We'll do all flavors. So just to simulate this, let's simulate it with the CG_flavor. Okay, so we're doing one iteration of this for loop now. We just want to see kind of what this stuff looks like in the real shell before we run it, right? So, okay, what is this going to do? Okay, this is going to construct one of those comma-separated commands, command line arguments that the thing needs. So let me show you what that looks like, and then we can unpack how it's done. That is the sample names comma-separated, and that comes out of our key file. So how does this work? Well, I used a little awk script. The awk is reading, well, first of all, this $paren thing is how you capture the output of a shell pipeline into a shell variable, right? Okay, so that expands into the output of the command, which we then assign to a shell variable. Awk, I'm running awk as the command. Awk is getting its input from the key file. And awk runs this little awk script. Hopefully you talked about awk scripts in your intro to Unix thing or something. But awk is another little mini scripting language, and it's just convenient when the program is so simple that it fits in like a command line argument as it does here. And there's even shorter ways to write this in awk, but this is sort of the style I like. For each line, that's what these curly braces mean. For each line, we run the following code of the input file. And we print out some stuff. We print out 2 things concatenated next to each other. The first thing is a comma separator that deals with the comma separator business. I'll explain that in a second. And the second thing is just the stuff in the first column. Remember, our file has 2 columns, and the first column is the sample name. And it's a tab separated file, so this tells awk that fields are determined by tabs. The colon backslash, the $quote_t isn't really an awkism, it's really a bashism. What that expands to is a tab. So that turns into a literal tab. Now, because of tab completion, you can't type a literal tab into your command. If I type x tab, no, how do I get a tab into the argument? That's what kind of this really wacky escape sequence is for. That $quote activates kind of a really unusual form of quoting in bash, and the \t inside that kind of quoting means tab. That's how you sneak a tab into an actual command line argument when you can't even type them. because it's taken over by tab completion. Okay, so that basically just tells awk that -f says change your field separator for whatever this is, and that bash turns that into a tab, and then awk just gets an actual tab. That's how that business works. So yeah, $1, this is the first tab separated column. How do I get the comma separators right? Because I don't want this, right? Oh, sorry, the Control C is confusing you maybe. I don't want that. I don't want that. I don't want that. I want that, right? I only want the comma between the things, not at the beginning, not at the end. So an easy way to do that in awk is that nr is a magic awk variable that tells you what line number you're on of the input file. And then $:colon is a construct that actually comes from the C programming language. And it's basically if the first thing, then second thing, else third thing, right? That's basically, it's the same thing as first $:second, third. It's just this is the expression compact form for that. And so if we're on the first line, nothing, otherwise a comma. And that does exactly what we need, right? So on the first line, because that goes before the sample name. So on the first line, we don't do anything. Then we do sample name. Second line, then we do a comma, sample name, and so on. Last line, comma, sample name, and no comma at the end. So that's how you get the commas just in between and not at the front and back, right? Sometimes people want terminators. That means there would be an extra one at the end. Usually that's the two major kinds. Separators, which are only internal, or terminators, where you would have another one at the end. And then the help for BS-Bolt aggregate matrix says separators. So that's what we're doing. Okay, so the next line is exactly the same analogous thing, but for the file names, which are also much nastier and so on, so because it's going to be ugly. But the how that's computed in the script is exactly the same. It's there, and if we tr the commas to new lines, at least you can kind of see that it's right, and then the commas are in there, right? So that's all well and good. And then we got to build this BS-Bolt command line, and at some point, making all of your shell script lines get ridiculously wide is also problematic. So sometimes I just use shell variables as ways to build up what would have been really long lines in the shell script. So options is the start of the command line options to the BS-Bolt aggregate matrix. So there's our dash verbose, dash T2 cores, there's our dash sample names, there's our dash file names. And then we're going to actually add some more to that. So we set options to be options followed by some more stuff. We're just making it longer, but I didn't want the line of text in my script to kind of get longer. So it's just a way of kind of line wrapping in the script, really. Now we've added the min coverage for the min sample 58333 business, which isn't quite right yet because we haven't changed the minus to a plus yet to work around the bug. That'll be on the next page. And I want to embed some of these parameters into the output file names like we've been doing as part of kind of breadcrumbs, as I call them. And so that's going to be a little thing that we're going to embed into the file names. We're working on the CG context. Min coverage is 4, min samps is 4. That is the two important ones to really record in the file names besides the context record. Okay, so now we're getting pretty close, right? So I'm not going to run BS-Bolt aggregate matrix. Let me just take everything from that point onward and just echo it. Rather than BS-Bolt it, I'll just echo it. So I can at least see kind of what the command line arguments were, right? All right, so there they are. And maybe if I want to see each one in its own line, I'll change spaces to new lines, and sort of then the arguments will be each on their own line, just to sort of preview this. So there it is. First argument, aggregate matrix, dash count, dash verbose, dash T2, dash S, blah, dash F, blah, min coverage 4, min sample bud, dash O, and here's the output file name. I'm going to call plex 6, like we talked about that. I'm not going to encode the sample names in there. And here we're going to suffix that CG, min coverage 4, min temps 4. And we're going to add TSV at the end for tab separated text. You know, if you download that to Excel and open it up, that would make sense to Excel. As an extension for the hint what the how to read. That looks good. All right, that looks good. So we have fairly high confidence here that this script is going to work. Okay, so we're just going to run it now. Okay, so I put that in stage 6 or 7 BS-Bolt ag for aggregate aggregation. The real script has all this extra text, and it's for all these reasons we talked So it's just to say warrant. Oh, and here's another little thing. I don't like this. There's no reason for BS-Bolt to do this. It doesn't really harm us because the way we use it in R, it doesn't matter. But BS-Bolt puts the output file, it just chooses its own order for the columns. I don't see any reason why you would do anything other than in the order that I told you the samples. Why don't you give me the output file in the same order? Why are you going to permute them into some order you like better? But it does, right? And I don't know the rhyme or reason really. If you leave the source code, I'm sure it'll become clear what it's doing inside. Maybe it's using a hash map or something. Who knows? There's something inside there is reordering the columns, and it doesn't take care to put them back in the order that the user put them on. So as I say, not really a problem. It does give you a header line of output with the column names in there, so we'll have to use those to figure out which column is which. And the way we use it in R will make that very natural. But it could be a surprise, right? It was a surprise to me, right? I mean, why would you do that? Why would you do anything other than give me it in the order I asked for? So it's just a thing to be aware of. Here's the whole business about fixing that thresholding bug for minimum number of samples. Oops. Wait a second. Lost my place. Yeah. Here. Yeah, we're about to run. Okay. Then, as I say, just a reminder, I think it's supposed to guarantee that the output is in sorted order by group by chromosome, ascending by coordinate within chromosome. So we're not going to do our own sort here. That's documented. If it didn't do it, it's a bug. Yeah, I did check the outputs in a couple cases, and they were good. And finally, that it just a comment that it does not make a gzipped output, which is okay for us. The files are really not even, I mean, they've still got some size even for us, and for a big run, they would be big, and I would keep them gzipped. But it, for some reason, unlike all the other modules, doesn't seem to produce gzipped output. Okay, so now we can run it. Why don't you guys run it, right? So this one doesn't need arguments because it's the merger of all the files and stuff, and it just grinds, right? We don't have to tell it to do each thing individually or something. This is the merger of them all. So there's no parameters to this thing. We just run it. Okay. So there's a little progress bar starting up, and it's very likely we'll see another little minor bug happen in that. Good convention for programs that produce progress bars, and you can see how sparsely it's kind of updating this, and it looks like it's updating once per sample, right? That's the 6 out of 6, right? So this progress bar is sort of one chunk per sample. You kind of wish for finer grain than that, but such as it is. But you can also see there's something about like how much time is allowed to elapse before it updates the progress bar, and at least a couple times that I've run it, we'll see if it reliably happens here. You know, we've got the methylation, we have the methylation fraction version, the count version, and the uncount version, right? So there's the methylation fraction version, and then the 1 column per sample output, the 2 column per sample output. So 2 flavors, 3 contexts. We're running it 6 times. It looks like we got four runs. I'm sorry, there's 2 runs done there because it seems to have 2 stages for each run. It's finished the CG 2 flavors now. Now it's working on the CHG flavor for the count flavor. Okay, so the file names have a little count embedded in them or an FR meth for fraction methylation to indicate which flavor of output they are. So we're now, we got, I said, 3 contexts, 2 flavors each, 6 runs total. We've got 2 done. We're working on the 3rd. Pretty reliably for me in here somewhere, there's a progress bar that doesn't look like it finishes, like it just stops in the middle and never updates. The program is fine and it ran okay. And usually when you write programs that generate progress bars, if the progress bar doesn't kind of update continuously or something, if it's sort of incremental with delays or something, you guarantee that when you finish your loop, you force the progress bar to be at the end because the user, it's not good to leave them hanging and like see like a half completed progress bar when you're actually done, and then they wonder, well, what happened? So they left out that line of code that just says like, oh yeah, we finished the loop, and so progress bar should be forced to 100% update. So it's just a, you know, it doesn't affect anything. It's just a visual thing. Details. All right. So you guys, it's run successfully for you guys? Somebody? it's grinding away. We could actually start looking at the output files. If you open a second Hoffman window, we can poke around and look at the output files while it's still making them. Since we're not running this in the job submission, the primary disadvantage, I suppose, is the fact that our session's busy while this is running, so we can't go exploring around while this is running. There is a way to do jobs in the background interactively, but I didn't, we get into so much, I didn't want to add yet another kind of shellism to get into that kind of would be take a little bit to really explain how background, foreground job control works in the shell. So yeah, we could have run this in a way, we would forgo the progress bar, but we could have run this in a way that would have given us our shell prompt back while this thing worked, even though we're running it interactively. But we just didn't do that. We rather had the progress bar. So, but maybe since I'll start another window just so we can start poking around while this thing is, because this is going to take like 15. I think the CHH takes quite a bit longer, so I think we're 10 minutes away still for the full business, and it's CHH is the big one. We got the CEG, both 2 flavors, and CHG 2 flavors done. It's now CHG second flavor is almost done. But yeah, that CHH is going to take a bit, because remember, the human has so many more CHG sites than CG sites, and so many more CHH sites than CHG sites. Okay, so share screen, and we need terminals, keyness. Right. Okay, so now we've got this. Oops. Now we've got this. Oh, crap. That's the one I shared, isn't it? Right. So that was the wrong command. So yep. Maybe I should use larger. Probably all going blind for the size of the text. Okay, so all right. Okay, can you guys see this window? Have I shared the right window? So you can see the one in the back that's still working with the aggregation, and then there's this new window in the front where we're going to poke around while that's still running. Somebody tell me, can you see this additional window? Yes. Okay, great. So here our new files are arriving, right? These plex 6s. Okay, this is the output of this aggregation script. Let's look at the CG's done. Let's look at the CG. All right, so this is the CG flavor that is up, right? There's two flavors, right? So this is the count flavor, and down here is the CG fraction methylation flavor. Okay, let's look at the fraction methylation one first, because that's less common. All right, so you see exactly what I was kind of telling you about. Okay, so we've got the site, which the way the BS aggregation expresses it is chromosome name : numerical coordinate. It mushes these into one column. In a way that's convenient for some of the stuff we do in R, not convenient for other stuff we do in R, and not convenient for Methylation. But it's easy. If we need to get this back into 2 columns and change the colon to a tab to split it out into 2 columns, we can do that. That's if it was 2 columns, we needed to go to 1, we could do that too. It just had to make a decision about which one we're going to do. The other one is easy to convert to. So the chromosome : coordinate is packed into first column. And then we have 6 samples. 1, 2, 3, 4, 5, 6. There are the titles. You can see they are not in the order I asked for, because I certainly asked for rep 1s before rep 2s, right? The tissues are paired, and though, I don't remember, is this the right order I asked for? I think I did the endoderm, mesenchyme, neural progenitors. I think that's the order I asked for, but why these 2 swap places, I don't know, but such as it is. And they're just fraction methylations, right? This is that 6th column from the parent input CG map file. And now you can see there's also missing data, because we allowed up to 2 guys to be missing, right? So some rows should have no missing data, right? So 1, 2, 3, 4, 5, 6, that guy is complete. With, we had our threshold of what? It was like 4 coverage or something. That's the coverage we had. Some guys will be missing 1. Some guys, yeah, there's a guy missing 1, right? And the way it indicates missing data is NaN, okay, NaN. not a number. The IEEE 74 floating point has a reserved pattern for many reserved patterns for not a number. It's typically used as the missing data in C-like programs. In R, you're probably familiar with NA, which is R's favorite missing data marker. And when we import this file, we'll turn these from NaN into a 3-letter string NaN. We'll turn them into real RNAs, so that all of R's missing data business works kind of like as expected. And you see some lines have 2 NaNs, which is it. But nobody has got more than them. Okay. And if you want a really hacky way, we can check that. All right, the first line is the first line. I don't think there's any lowercase a's up in there. No, don't think so. And this is numeric for us. Everything is numeric. The nans are nans, and each nan contains a single lowercase a. So here we're just going to do something really hacky here. So we're gonna tr, and if you read the tr man page, you can flip the sense complement. -C flips the sense. So tr complement d a and new lines, meaning delete everything but little a's and new lines. Okay. And in the file I want you to do that is the CG map fraction, the one where we're just looking at this. Okay, blah, and then less that output. The number of A's you see is now the number of nans that was in that row. Okay. All right. So let's sort it, and then unique -C. There's the histogram of how many lines have how many nans. All right. Not a real robust way of doing it, but sufficient for our purposes. Okay. So we see 0, 1, and 2 missing, and it's exactly the threshold we asked for. So working as designed. But I tell you, if you did it with the number where it's supposed to be, the fraction where it's supposed to be, then it comes out that you also get 3's. So, but this is working as expected. Okay, so now let's look at the count flavor. This is the CG context count flavor, and this is what this guy looks like. Now this has got more columns, right? Okay, so luckily our window is wide enough that actually the data doesn't seem to wrap much, but it's hard to read this header line at the top. We'll check that out in a second. We'll head -N1 it out, and then we'll change the tabs to new lines, and see just a better way to verify that's what we're thinking. All right, so otherwise it's the same thing. So we got the chromosome colon coordinate, and then at least you can look at the first 2 columns. So that's mesendoderm rep2 and mesendoderm rep2. It's 2 columns, and then it just underscored this meth cytosine, that's the numerator, and the total cytosine, that's the denominator. So 7 out of 7. Now why BS-Bolt chose to put .0s in all these that are integers and just waste space in the file and make it seem like it's a floating point number when it's really an integer? Don't. Right? Another easy fix to the code. Probably another thing I should mention to the guys. But there we go. Like 7 out of 7, 11 out of 11, 39 out of 47 for this one, right? That's the third tissue, whatever that is, right? So that's the third sample. So here's first sample, second sample. So mesenchyme rep 1 is 39 out of 47 in first place. All right, anyway, so this is useful because it's equivalent to the fraction meth file. Just take this and divide by this. But it also gives us the coverage information, which is important too. So this is the file we actually like more. When we import it into R, this is the one we like more. All right, so how's this guy going in the background here? So we've got 1, 2, 3, 4, 5 of them run. Oh gosh, there goes my A key. It disturbs our beautiful progress bar. Maybe it'll recover. Yeah, the A key just sort of takes off on its own sometimes now. But okay, so CGs are done, CHGs are done, and one flavor of CHH is done, and we are waiting on the last flavor of CHH. Now I have half a mind to just Control-C this thing and quit it because we don't really need the CHH, and maybe in the interest of time. There's our progress bar. Progress bar recovered. Okay. All right. What's the estimate for finish on this? This is the last one, second stage. We only got 1 sample left to do, which is like 20 seconds or something. Okay, maybe we'll just wait it out. Have a quick break here. In in software companies, the programmers, you'll find them half the day, using their Nerf guns, shooting each other, and you're saying, "Oh, I'm compiling," whatever. It's you're waiting for the machine to do work, right? It could, of course, do something else, but waiting for compile. Right? We're waiting for analysis. We'll do other things. Oh, I thought it was on the second stage. That's a side effect of my typing the A's, the machine typing the A's, and causing me to have 2 progress bar lines for only the first stage. Okay, so now we're starting the second stage of the last round. It's still like a minute estimate or something. All right, patience. This is a Zen bioinformatics break. You can do it. 2 samples. It's like boiling water, right? It's slower when you watch it. Okay. Clearly, BS-Build is also a fan of the progress bars that reach 100% when not quite done. There we go. All right. Okay. Now we have got a beautiful set of these Plex 6 files. Okay, so now we've got the quantification, the primary analysis sort of rearranged in a format that's more easy to use in like R. So okay, so we did this. We took a peek at the files. Oh yeah, I was going to show you that the column names in the bigger one are kind of what we expect. So head-1 and 1 gives us the first line, and change tabs to new lines. That'll just, those are the column names, whatever. You want to see what the awk NF does, just to verify that. Let's add a new column that has the NRs, the record number. And there you go. If you needed a little key to the columns, right? Column number 1, site, 2, whatever. Anyway, that looks good. We've got 2 columns for each one. They're named in a completely regular way and so on. Everything is easy. Okay. So we need to download these to our local machine. And because we're going to use R on your local machine, hopefully you've all got your favorite R environment, R Studio, whatever you like to use. So I tend to use base R, the RGUI app for Mac OS. It's the base one. It's good enough for me. And so we need to get the files to your local machine because we got to the R. I want to use R on the local machine because we're going to make plots and stuff. And we can do that on Hoffman, but I don't want to get into that. It's easier to work locally, and we're not really doing any super heavy lifting in R. So running on our local machine is going to be fine. So I've got this extra window here, and I'm going to go to my desktop day 3, which is a directory I don't think I made yet. So okay, desktop day 3, make dir, change to. All right, so here we are. Yeah, okay, so clear that off. I am in my GUI, this is my local machine, Hades. This is my desktop day 3 subdirectory. This is where I want to put the stuff from today that I want to copy out. So here is the SCP command to copy it, recursive copy. You got to change this to your username. This is your scratch space. And we're just going to grab this whole, oh, I haven't made that directory yet, right? Oh, right. So usually we clean up, right? This is done now. This run is done. So I'm going to make a stage, this is the name I want to give this directory for this stuff. So I just made a directory to put these files. unlike most analyses that we did, which put their outputs in the same directory as their inputs, the way we wrote this script, in a way it makes sense because the way the aggregation works, it dumped its output in the same level as the script and not in the place it pulled the CG maps from, which would have been this directory. So they're just sitting on up here next to us up here. And they're the only thing with a capital P. So I'm going to grab all the capital P stuff and put it in this new directory. Oh, I forgot the VI. But there they go. And so I've now got the stage 7 guy with the outputs from this thing here. And if I, maybe I should have logged the output of this thing and copy pasted this into a log file. You know how that goes now. I don't really need to really demonstrate it over and over. Okay, so yeah, so that's ready. That's already in place. And I'm going to FCP recursive this directory down to my local machine in the place where I'm at. So here we go. Okay, so that's going to copy that. Well, we should probably check how big those were. du-h, it's 361 megabytes. That is the size of the individual files, and the big ones are the CHHs, of course, no surprise. It's only 13 and 12 meg for the CG guys. And like I said, you don't even really need the fraction methylation ones because they're so trivially computable from the count ones. So really just need this 13 meg CG file. That's really the only one we need. Okay, so I'm now on my local machine in this directory, and I've got my files here. Okay, and now I'm going to start R. Okay, I want to kind of be fancy. I can do it that way rather than dig it out of the GUI. And I've opened up a new window which you can't see yet. So, okay, screen sharing stop, screen sharing start. I checked with my wife. She said she doesn't know of any way to add windows to screen sharing. R terminal, keynote. There we go. Okay, so R. Okay. Now, can you guys see this R window? Is that showing up? You're probably in R Studio or something, but somebody tell me if you can see this guy. Yes. 1014. Yeah, that's current. Okay. Okay. First thing I need to do is get R into the place where my files are. Okay, so I just set my working directory. That is essentially the CWD, the CD command in Bash shell, change directory, is called setwd in R. Okay, and so I just moved into that place. And in R, there's ways to get help and stuff, right? So that opened another help window, which I'm not sure whether the share is everything in R or just this one window. But I've got this help window here, and this is how it's the help window for getwd and setwd, and it actually reminds me that list.files will tell you what the files are in the place you are here. So anyway, you can kind of see we're in the right place. Okay, so all right. In R Studio, I think there's a little GUI thingy you can click somewhere and do it, but it's going to run the same command inside. Okay, so that's keynote. Okay, and because these file names get long, I'm going to make an R variable that just contains a long part of the file name. And hopefully you know the paste 0 command in R. That is how you put strings together in R. So paste 0 takes strings and puts them together. The reason why they called it paste 0 is because the one that was already called paste uses a space between them, which is often less useful. So that's why I called paste 0. Okay, you probably know the read table command. Okay, so that's what we're going to use. Oops, sloppy with the selection. Okay, read table, and I took that biggest part of the file name, added the rest of the file name. I'm using the CG map, the CG context fraction meth version to start with, which is the single column version. And what options we need to read table? Well, it's got a header line, which is nice, because then that'll turn into column names inside R. The class of this thing that we got out, this is an R data frame that came out of that. And data frames got those labels on the rows and columns, and that'll magically take the header line and turn it into the column labels. And row names equals 1 means that actually the file contains the names I would like to use for the rows in the first column. So that activates that. So now our data frame has the row and column names all set up. It's a tab separated file. and I want you to turn the NaN strings in there into the RNA, right? So R is native, not found is missing data is NA. Although R actually also has, I thought you had NaN. Oh, because you call it NaN with a capital N and a capital N. Yeah. Anyway, the default NA is a logical and the NaN is a numerical thing. So R kind of has both, and it actually understands both of them as missing. They mostly work. And we could have imported it as a capital N, but more traditional missing data value file is the capital N, capital A. So that's what we imported as. Okay, so let's see what the head of this thing looks like, right? So you probably know the head is how you can look at a big thing without having your screen explode. So here is the should show us all the columns, but not just the first few rows, right? That's what the head does. And so you see it imported exactly as we saw it in the shell, right? So there's our 6 columns for our 6 samples in the same order as the file. And here's our row numbers, which I took as the sites names, the site locations. And then here's our data, and we've turned those names into RNAs. Everything looks great. Okay, and how big is that? That is a 201,671 by 6 data frame. Okay, so that looks good. Now, I'd like to make some plots. These plots are all going to be repetitive because I want to do them for all combinations of 6 tissues and so on. I don't want to type the same histogram command with all the options over and over again. So one pattern I often do in R is to make a temporary throwaway function that's only going to live for a very, very short duration. It takes some arguments, which is the part of the command that varies, and then the rest of it does the stuff that doesn't change using those variable parameters. And then I'll just call that function do or something, and then I can just do a whole bunch of do's down here, and then the repetitive stuff I don't have to keep repeating. Par MF row, you may know, is a little bit of magic that means that the next figure you generate is going to start a grid of figures, okay? So here I'm asking for a 2 by 3 grid of figures. And as I generate figures, for example, with the histogram command, they're just going to populate in the order, okay? So they populate in the order of same way English goes, right? So upper left corner, go across the first row, then back to the left, go across, and so on. It fills it up like that. Okay, so what I want to apply, I just, this is one of the things we talked about. I showed a figure on day 1 that's like what a typical CG distribution looks like, a histogram over all sites, right? And it's like concentrated at 0 and 1, and there's little bits in the middle, but the middle is 50% is kind of empty and so on. Let's look and see what our actual data is for these, right? So we are going to grab from the table, the data frame we just made. Like this is the column for mesendoderm replicate 1. So those are fraction methylations, right, that are in that column. And then we want to give the plots names so we know which one's which. And so that's the mesendoderm replicate 1. And so the function takes the vector of fraction methylations in the first parameter and the name of the second parameter, and we call the histogram command. And for some reason, yeah, the indenting should be, oh, maybe I did that because it starts to run into the plot. Move this over a little bit. There we go, that's prettier. So hist-vec, that's all the same. I would like all these histograms to not use the auto detection for where to draw the breaks. I want them all to be consistent with each other. So this thing will do 0 to, I'll express it, it's a fraction. So it's 0 to 1 by 1.01, meaning 0% to 100% by 1%, right? But it's expressed, this whole thing is expressed as fractions. So, right, so that seq thing just generates that list, right, from 2-step, right? That's the way seq goes. It's a very commonly used thing in R. And the C parentheses stuff is how you build lists in R, right? So that C01 is really just a list with 2 entries of 0 and 1. Right? That's basically how you give a pair, or a tuple to something. That's very common in R too. So I want to force the x-axis limits to also be 0 to 1 because I want all these plots to be kind of the same, right? So they're more easily comparable. I'll let the y-axis float. Okay, I'll just assume it's going to do something. Okay, we could force that to something fixed too if we had to. I think the default is like black outlines, white insides or something. Anyway, how about dark green frame with dark green inside, solid dark green bars basically. The title of the plot is the second argument. The X label for the X-axis is fractional methylation. The Y label is number of CG sites. And then we just call this thing 6 times for our 6 replicates. Okay, so and bada bing. Okay, I don't like that aspect ratio, so we could put that in code, but let's just GUI mush it around. Okay, so my next question is, can you guys see this window, or are you going to be able to see all my R windows, or is it just sort of the screen sharing is stuck to like this one R window? Can you see this figure? Oh, you can't see the plots. But if you're following, okay, so I don't know if you're following. Have you guys got your CGs generated? You can always Control C the thing running and not make the CGs and CHGs. Have you guys got the CG files? You got the CG files and downloaded locally, and you're able to follow this along in R and start making your own plots. I know. Actually, I'm not sure how to pronounce your name, so I don't know if it's Steen or.
Yeah, it's Steen.
Yeah. Okay. Great. Steen says he's got it working. Anybody else got there? Or just tell me what stage you're at, like downloading in R but haven't got there yet, still generating on Hoffman, or what stages are you guys at? Okay, Amanda says she's got the plots. It's 2 people that are making plots. Anybody else following along? I guess that's half. So, all right. So yeah, so that looks good. So now I've done a fairly fine binning here, 1%, and you may wonder why. You can see that like the mesenchyme actually looks kind of okay, right? I mean, it's kind of mostly methylated, and beyond that, mostly unmethylated completely, with fairly little in the middle. But the reason I wanted to really do a fine binning is because our data is discrete enough that you really kind of want to see, like if our coverage is 4, you could get like 2 out of 4, and that's 50%. I mean, you can only get 0 out of 4, 1 out of 4, 2 out of 4, 3 out of 4. So I kind of wanted to see if we have a lot of big concentrations at very small integer fractions, right? Which would kind of suggest that if you do your binning very coarsely, then that kind of hides that, and you may get a big bump in the middle or something, but it's really just the 1 out of 2s, and maybe you haven't filtered for denominators enough yet or so on. So it's useful to kind of start with a fine binning just to see if you've got any sort of discrete integer problems. And these don't look too bad, actually. We don't see like a big spike at a half. There's probably a little blip at a half. It's related to low coverage, but these look not too bad. Now that we've seen them like this, we might decrease the bin size, right? So we could go and edit these breaks here and change this to 5.1, and then rerun this, right? And then that'll give us guys with every 10% instead of every 1%. And if you run that yourself, you should have a plot that's probably more comparable to something you might see in a paper. These are not deciles. But now that we've verified that the discrete fractions aren't really killing us, then that's what this looks like. So, I mean, this looks, actually the samples look a little different, right? Mesendoderm seems to have a little bigger tail on the high end, and the low points for these guys seems to be more around 1/3 and not at 1/2. And the samples do look a little different, right? So like mesenchyme does look more binary than, say, mesendoderm. And even the neural presenters look a little different in this histogram. But anyway, we're making plots. Okay, so why don't we grab the other file? Because actually, to do much more now, we kind of need the coverages. So let's switch from this fraction meth file to the coverage-based file. So we're doing another read table, but with the coverage version, the numerator-denominator version of this file. So this is the one that's got twice as many columns. and it's all integers with the numerator denominators that we would formally look at the fractions. So first thing to do is let's mush this thing up into something that's nicer. These column labels are kind of horrible, and we'd like to have all the data available all at once. So that means I would like the ratios, the fraction methylations in there. I don't really need the numerators. I'd like to turn the numerators into the fractions, basically. Keep the denominators, that's basically coverage, rename it coverage. And I'd also like to pool by replicates, right? So I'd like to keep replicate 1, replicate 2, and then the pool, right? So let's rearrange this business, okay? So, and also to do like some of the plots later, I also need those chromosomal coordinates as numbers, not as strings mushed up with the chromosome name. So here's a whole bunch of transformations we're gonna do, okay? So start with these 3 at the top here. And locations is the original names with the merged as a string with the column. Then there's the chromosome numbers, which are just the 21s. It's all the same for us, but I'm doing this, simulating this as if you had a file that was for the whole genome. So that would be the chromosome that each entry is about. The chords are the coordinates extracted out and actually turned into integers. So that is useful for how we're going to, that's good to actually work with them, right? So that's an integer vector basically. So that looks good. That's been rewritten. That's useful. Okay, that started us on this path. That's kind of like the row labels. We'll make a little temporary function here because we'd like to turn those NAs to zeros, right? So for the coverage, numerator denominator, we might as well turn the NAs into zeros because 0 out of 0 is basically no observation, right? And in R, 0 out of 0 turns into nan anyway, which is perfectly usable as a missing value. And coverage 0 also makes sense, right? If we were adding up coverage or averaging coverage or something, anyway, the 0 would make sense. So let's turn those NAs to zeros, and then we won't have NAs. We'll have NAs in our fraction column when we do 0 over 0. That's okay. That'll represent a missing value in the fractions. And actually, we're going to use percents rather than fractions because the divisional display fractions not so much. Percent makes more sense for humans. And so, yeah, let's turn those NAs to 0. So this little NA to 0 function, if you have a little list, let's say 3, 5, 7, 9, NA, 9, 4, 2, 5, nan. Let's see if this works. Okay, so list is this. Yeah, that's the actual code. All right, LST. Actually, that's the code for LS. I lost the T and it showed me the source code for the LS R function. Okay, anyway. Here's my little list, and if I run my little NA to 0 on this guy, what comes out? 3, 6, 5, 4, 0, 9, 2, 4, 6, 0, 9, 2, 4. It works great. Okay, so that's a little guy that we can feed in vectors, and it'll give us NAs replaced with zeros. And then we've got all these columns to do. We've got numerators and denominators for all 6 samples to pull out. And I'm going to really abbreviate these variable names, right? So we've got mesendoderm, ME, that works good. We got mesenchyme, which I'll do MS, and we got neural progenitor, so I'll do NE. Okay, so it's memes and me, right? Okay, it's about right. Replicate 1 and 2, capital N for numerators, capital D for denominators, little p will be for percents. And so this is the business, right? So basically, most of these are just extracting out the column and turning the NAs to zeros. Then we can compute the percents as numerator over denominator times 100. Okay, so let's do all those. Okay, bang, 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 bang. Okay, we've got all those variables ready. All right, so if we look, there's a numerator guy, there's a denominator guy, and there is some percents, right? Now it's like 100, 140, 14. Okay, looks good, looking good. Now let's do the pooling of the replicates within tissues. Okay, so we got 3 of these to do. We need numerator, denominator, and percent, and I'll just take away the 1 or 2. All right, so ME, capital N, is now the numerator for pooling the mesenchyme, sorry, the mesendoderm. Okay, so that is ME1N plus ME2N, and the denominator is the ME1D plus ME2D, and the percent. So we actually have a choice here of whether we would average the percents or merge the numerators, merge the denominators, then do the division. Those are different, right? So the latter way kind of gives more weight to the guy that has more coverage. The other way just averages the percent. What I generally would recommend, I think it works out best usually in practice, is to, at the site level, it's okay to merge numerators and denominators, and it's probably what you want. When you compare different sites, you generally don't want to do that. You really want to turn each site into a percent and then average over those, because then each site gets equal weighting. And if you have some guy in there that's got really high repeat coverage or something, and he expands, and he'll take over, right? Because if you do kind of the add numerators divided by add denominators, then the guy with most, if some guy has, if everybody's got coverage 5 and somebody's got coverage 500, the guy with coverage 500 sort of takes over, right? Pulls that really hard in this direction. Whereas if you turn them into fractions and average those, then that guy that would otherwise pull very hard is only one nth of the sample, just the same as everybody else next to him. So anyway, so that's why I kind of recommend at the site level, maybe pool numerator denominator, then divide, but when cross site, then divide, then average. That's my recommendation. Okay, so we're going to basically, these are at the site level, so we're going to do the pool numerator, pool denominator, then divide method for this. Okay, so there we go. And these look just the same as the other. The coverage will be a little higher now because we've done some pooling. 4 plus. Oh yeah. So if we wanted to verify, I guess we need the ME1D and the ME2D. That's the two denominators that we added in 28 plus 15. That should be the 43. Yeah. Okay. And then, so anyway, so the coverage has been pooled now. Now we could take a step further, and I'll leave that to you guys as an exercise. We could work on actually finding the CG sites that are next to each other, the CG plus strand minus strand. We could take those two sites and pool their data too, and double our coverage again, essentially. I won't do that here for the complication, but if you want an exercise for yourself, then that could be something you could work at. All right, we basically now got everything. We've got all the pieces for how we want to rewrite this thing, and now we'll just collect all the results in a big data frame. Okay, so that's called hold CG. And what does that look like? It's got all the juicy data all combined and ready to go. And we've got every combination we need to kind of do anything we want, right? So the row names are the strings merged positions. We've actually got a column separately for chromosome name. We've got a numeric column for the coordinate parsed out. We've got the percents for all 6 samples and all 3 tissues, pooled samples. And we've got the coverages for all 6 samples and all 3 tissues, pooling samples. Okay, so we've got column names that are short enough to type and easy. I mean, this is now, an efficiently usable guide, right? Okay, so this is much better than the raw import was. Okay, so all right. So yeah, so this is a juicy guy. Now, if you were doing whole genome, this guy has the information for the whole genome. Because I want to do some stuff later that's only chromosome specific, I'm going to subset this to a single chromosome of interest. It does nothing for us. But if you had whole CG as the whole genome guide, this would pass down to the subset that belongs to a single chromosome. So I'll just show the R code for that. So we are interested in chromosome 21. Notice not using the number here, it's a string, right? Some people would have CHR21 and then it's less confusing, but we have chromosome names for the numeric, for the chromosomes 1 to whatever. We've got them really as strings. So that's its name. It's a string really. And here's how you pass to the subset. Okay, we're going to call it per chrom CG. This is the CG context for the current chromosome. You'll probably be doing this in some loop or something because the per chromosome would go through them one at a time because you're interested in plots for all the chromosomes. So I didn't call it CHR21 or something. You want to have it. It's probably a variable inside a bigger loop. We don't have the outer loop, but anyway, that's why I named it that way. And it's just whole CG bracket. And then the first argument is just the places where that CHR name column matches what we want. And then we want to keep all the rows. So we pull out the, well, that's the row selector, right? That tells us which rows to keep. And then we want all the columns. So we just don't have any specification for what columns we want. We'll just keep them all. Okay, and for us, that looks exactly the same as the thing we did. It did nothing, like I said, but if we had the merged data in whole CG, this Kirchhoff CG would just be the single chromosome Kirchhoff. Okay, so there we go. We're ready to simulate that process of passing from homogene to partial. And now let's just make our histograms again using this data, right? You probably never would have bothered to load the fraction file. Right? This is just where you would have started. So how we would have produced the fraction methylation plots here. All right, I'm going to make a little story do function again. Histogram, somehow my indenting got lost. Here we go. Histogram, first argument breaks. Now it's percent, so we'll do 0 to 100 by 1. Force the x-axis to go from 0 to 100, dark green, dark green. There's the title, there's x-y-axis labels, 3 by 3 grid. I'm going to do a 3 by 3 grid now because now we got the pooled, right? We got the replicates, 6 samples plus the poolings through tissues. So now we might as well add another row and get the full 3 by 3 going here. So let's copy that and paste that, and you should have a nice set of plot, a nice plot. Now that's with the 1% binning, which, if we're pretending we didn't do the fraction methylation stuff earlier, then we didn't get to see this yet. So this is our first look at it, and we say, oh yeah, it doesn't look like that much discrete integer problems there. Yeah. So okay, so let's well, we were right there. So why don't we change that seq 0 to 100 by 1 to seq 0 to 100 by 10. There we go. And maybe you can't see this window, but maybe I can copy it as a PDF and paste it into my keynotes. There you go. You should be looking at something like this, right? This should replace this figure here, right? So there we are with deciles, is what we histogram. And so that's how you do that. And we could have done that for CHG context, CHH, whatever. I mean, you kind of see how it goes now. Let's start inspecting coverage. Okay, right? So we got these coverage vectors now. Why don't we just histogram those instead? Now, if you take out one of these coverages, right? So and look at it, the maxes should be near 999. They shouldn't go over now that we've fixed kind of the filtering. Oh, I know I'm confused. There we go. So if I ask for the min and the max of this coverage business, 871 is the max. And it's going to be so sparse out at that tail. I mean, we could plot it as a log on log scale. Maybe we could do that, right? So if we could histogram the base thing, and that's not too helpful because it's sort of got these big numbers in it, but they're very low frequency, and so it skews the x-axis. Okay, well, maybe we could do a log scale. Histogram log 10 of that thing. I keep forgetting you can't see these stupid windows, right? But okay, so then I gotta. Okay, so here is the basic. Give me a new slide. So I got place to just duplicate this side. Duplicate. Delete everything on the slide. Now I've got a place that I can paste stuff in. You can see. Okay, there's our working on these stupid screen sharing limitations. Okay. Right, so you can't, this histogram is not useful because it's skewed by those big numbers. Here is the log version. Right, so that looks, at least we can tell what's going on. We can make some sensible idea of where to cut it off. Well, it looks like certainly by 2, 10 to the 2 is 100. So certainly if we cut off everything but 100, we're not really losing much. So that's why I kind of did what I did in the okay, so now we need to go back to here. There we go. And that is why I kind of decided that things beyond 100, I just want to pin to 100. So we'll take everything above 100, make it 100, so we don't lose the count idea of how much of its stuff there is out there. But our histogram plot will then go up to coverage 100, and we won't just get it all squished down to the left. That's the interesting part. So okay, so the do function is going to take coverage as the input and the title, and this is a little parameter inside this little mini function just to give an easy place to change it. Coverage max 100. We find the places where this coverage vector exceeds that. We index into coverage at those places and assign them all the same value, which is coverage max. We just pinned the max to coverage max. We histogram that thing. R also, as you know, is a pass by value language. So the fact that all these guys are getting passed in here from our big whole CG, this function essentially gets copies. And so we're not harming the original data by playing with it this way. This is just a little scratch copy that this function gets. Another reason to actually do it in these sort of functions. They get local variables. The breaks will go from 0 to coverage max by 1. As long as coverage max is something, like 20, 30, 50, 100, that'll be fine. If it was 1,000, that'd probably be less good. But that'll work for that. We'll force the x-axis to all be the same for all these plots, 0 to coverage max. Instead, to distinguish from the other plots, instead of greens, we'll use black. There's black. There's the title. There's the labels. 3x3 grid, same business, but now we're passing the coverage vectors instead, and there we go. And this is what comes out. Okay, so that looks good. So anyway, you actually see that there's considerable differences among these. The mesenchyme has a broader spread, meaning that mesenchyme is more unevenly distributed. The sites that got sequenced in mesenchyme, some of the, it's got more zeros and more stuff at the higher end, right, compared to the other samples. So essentially, some regions sucked up the coverage that other regions would have gotten, right? There was some competition there, and the coverage was uneven. The neural one looks kind of okay. The mesendoderm maybe a little worse and lower. Anyway, these peaks are not great, right? So maybe by the time we're pulling neural, maybe that mode is, it's hard to see without making this bigger, but it looks like it might even be above 10. Of course, there's a spread, but some things are 20s, right? But by the time you get to 25, that's not too common. Mesendoderm's got this part that goes up to 40 and so on, but but it's got a lot of zeros too. So anyway, such is our coverage, right? Ideally, you don't have anybody hugging the left axis, right? In whole genome bisulfite, right? Ideally, nobody hugs the left axis, and your bump is actually isolated from the left in the 0. And you get a little bump out somewhere, and hopefully the tail doesn't go out too far, so that's coverage unevenness. So, ideally coverage would just be a spike at some number, right? That would be perfect. Otherwise, hopefully it would be a little spread, but it would be a narrow spread, and it would be apart from 0. But here we are. We're sort of in a low coverage regime. Where it's like most common coverage is 0 probably, and then there's 1, and then there's 2, and so on. It just goes down from there. Maybe this guy starts to pull. Maybe the neural, especially once you pull neural, he's pulled away from the. So that's good. He's on his way to actually, having some real coverage. If he doubled the depth of sequencing there, he'd be in good shape. These guys, individual replicates are not doing too good. Maybe starting to pull away here with the pooling of those two, but these could all benefit. Neural P is in the best shape of these for coverage. These guys could benefit from some more data. Probably the only alternative we have without resequencing is to try to do less cutting, and we made the cut it upstairs, right? We should be less. We'd have to try being less aggressive there and try to keep more of the data. And if you just want to see what it looks like to, at this point, you can start doing things like scatter plot samples against samples, and that would give you an idea of sort of the correlation of coverage between them. Like, are they putting their coverage at the same places? So here's a little density map, a little hex tile density map. You could do this as a straight up scatter plot, but there's so many dots in this region in the middle, you're not going to see what's going on. Although there's not really many dots out in here. So if you're concerned with the regions out here, a straight up scatter plot may actually be better. And you can see actually in this sample, certainly at these sites, this sample, these are individual samples. No, I did the pooled ones, right? So this is the pooled neuronals versus the mesenchymals. And you can see that it's who's on the y-axis. That's the mesenchymal. So at these certain sites, mesenchymal has a way, oh gosh, which way does this go? It has way higher coverage than the neurals. But there's not many sites like that. Most of the coverage is here. Actually, that's just true generally, right? So, yeah. So what happens is that the sites where mesenchymal does have coverage are higher than the neuronals, but at some point, it's probably hard to see here, but at some point you get this stuff down in here where the way that mesenchymal did that is by putting a whole bunch of guys towards 0. And there's the bright, these dots are like this. So it's like these black guys are hardly anything, and it's more like this blue stuff is where the stuff is. And there's the stuff down in here, and that's where mesenchymal is very low, and the neuronals are higher. So the neuronals are just more stable. Anyway. So you can play with all kinds of combinations there, right? So you may want to have coverage thresholds. We can re-threshold coverage now that maybe we decide 50 is the highest we want to go, and maybe we want to actually lower the minimum to 10. Well, now that we have the data in R, we don't have to regenerate the input files and all the filtering that way, and so we can do all the filtering in R, right? So we can go and pick out for each condition. And we could decide whether we wanted to take them all to the same subset or we want the subset to vary in each one or whatever. Here I've let the subset vary and be specialized to each one. But here for the Zendoderm ref1, let's take the places where that particular guy is between 10 and 50 in coverage. And then we'll pull out the percent methylations for him at those places. And so we've passed to the subset of places that for him he's covered 10 to 50 times. And the same for the rest here. So, you can copy and paste this in, and this will give you this sort of coverage subsetted specialized plots here. And you can actually see the pooling now. Maybe these histograms are sort of cleaning up now. They're looking a little more continuous and so on. It's because we're dealing with, denominators are a little more controlled now. We're losing sites to do this, but the denominators are kind of acting more reasonably. Okay, so that's kind of how that goes. Okay, so coming up on 2 hours. All right, here's a nice example, right? You see these all the time, right? So this is the chromosomal overview plots. Looks like Keynote's having a hard time rendering this thing because this thing has got like, maybe it's a couple 100,000 dots and there's transparency and there's sort of a low-res preview. There it goes. It kind of snapped it down. So these are chromosomal overviews. So what this is the whole chromosome 21 is on the x-axis and it's labeled nicely in megabase pairs. I mean, this is almost a publication-ready figure, right? We've got x-axis and megabase pairs. We've got y-axis is the percent CG methylation, and we've averaged in bins, right? And I think the script is currently set up for 10 KB bins with no overlap. Just every 10 KB we make a bin. And so we plot the average methylation in that bin, using the method of just taking the sites that have a percent methylation attached to them and average those percents, right? So this is the cross-site type of average I would recommend, right? You compute the percent at each site and then you average the percents or the fractions, whatever. And so we've got mesendoderm in the first row, we've got mesenchymals in the second row, we got neural Ps in the third row, and the replicates are the rightmost two columns and the pool is in the left. And you actually see that the pattern, I think, is stable across the replicates, and so we might as well just look only at the pool. And you see some interesting things. So I think there's chunks of this chromosome 21 that are gappy, right? So I think this is just, you remember when we looked at the FASTA file, it had all those ends at the front. I think those last like megabases or something, right? So there's two chunks which are probably poorly represented in the reference. They're just mostly ends or something. And then there's some sort of transition zone here, which is interesting. And in mesendoderm, it's fairly high. I mean, there's individual bins, 10 KB regions. I mean, they could be, little parts of promoters or who knows what genes, but it's mostly high and flat. And same thing with the neurones. But the mesenchymal looks quite different, right? This is a very different pattern. There's sort of regions at chromosome scale that look to be hypomethylated, right? Hypo-CG methylated. So that's already quite interesting. And people make these kind of chromosomal overview plots a lot. All right, so how we can do it? This is quite a transformation of the data to do. So the package I recommend to make this easy is the depiler. This is part of the tidyverse, so-called tidyverse of R packages, which is really convenient for manipulating data frames and so on in ways that are sophisticated but easy to specify. You can do this with base R, but you don't want to. So yeah, so get depiler installed in your package manager, however you do so, and then we can activate it with library depiler. So, oh gosh, why am I pasting it into a shell window? That's no good. All right, so there we are. Yeah, some symbols are getting played with. That's okay. All right, so to make the script parameterizable, base pairs per bin, 10,000, maybe to 100,000 would be a little easier, less dots to plot, and I think we get about the same information. Okay, so 100,000, 100 KB bins, and how do we do it? So this is going to become the thing that we plot. So you'll see what this is at the end, but it's basically going to have, for each condition, it's going to have the summarized data in the bin, right? So we got to rebin these things, and we've got individual chromosomal coordinates around. We got to rebin it, but this thing is going to collect all the plotting data. So it's going to be organized by bin basically as the rows, and we've got the samples across, and the samples aren't going to have the point fraction so on, but some kind of average fractions in each column. And that's what we're heading towards. So how do we do it? So we start with, of course, with the Kerr-Crom CG, right? So that is the data that's going in. That's the whole genome, you know, kind of guy that summarizes everything we could ever want. Right? So that's the thing we carefully set up earlier. Then you activate depiler. This is actually, I mean, it's intended to be read as some sort of fancy arrow that reads left to right. So it's sort of, you're doing some sort of processing to the thing on the left to produce a new thing on the right. And then you can chain these things, right? So we got a little chain here. We got another one in the next line and so on. And this is really just an operator that's got a horribly funny name. One of the things about R is that actually pretty much anything that looks like line noise, you bang random symbols together, it's a valid R symbol name. And so you can associate code with those and make them operators and so on. It's kind of cool. So, so this basically, this is really a function inside depiler. They're just doing this to make the syntax kind of, this is some syntactic sugar, really. And so first thing we need to do is to compute the bin, right? We're going to number the bins like 0, 1, 2, 3, and so on. Bin 0 will be the first 100 KB, bin 1 will be the next 100 KB, bin 3, and so on. So that's a little bit of the bin numbering. So we first need to turn each coordinate that each data observation is, right? Each observation row right now is currently associated with a point coordinate. We need to know which bin each of those belongs to. So how could we compute that? Well, we can get more magic about depiler is you don't have to keep saying Kerr-Crom CG $ to pull out columns of the thing. It sort of knows once you're starting with a table, of course he's referring to columns inside this table. Why would you make him repeat the name of the table over and over? So the fact that I can just say chord here now, depiler will know that it means, oh, he's talking about this thing. Okay, so that pulls out this column. And then we divide it by BP per bin. So we just divided all those by 100,000. Now it'll be a floating point number. But the integer part of it is the bin number, right? So because 0 to 100,000, you know, that'll turn into 0.0 to 1.0s, and then the next 100,000 turn into 1.0s, 2.0s, and 1.5 is the middle of that bin, and so on. So these give us floats, which are basically the bin numbers, but they got the fractional part, which we don't care about. So then the floor function throws away the fractional part by rounding down, okay? So that is literally an integer bin number now, okay? And we are going to create a new column in this data frame called bin. That is the result of that, right? So this is the thing we just computed, right? Which is the bin numbers. There we go. And if I just do head of that, oh yeah, of course. But if I do it this way, I got to say what table it comes from because regular R doesn't know what's going on. So there we go. That's the table that's got that guy. Right. There are bin numbers, 50, 51, 51, 52. All right, in the head function, how do you get more? I think there's got to be a way to get more. It's just, the second argument is just how many entries you want. Right? So give me some more. Right, okay, all right, give me a lot more. There you go. So you start to see, you know, the file is sorted by coordinates. You know, we guaranteed that. So, you know, the bin numbers are going to slowly ascend, right? But there we go. So there's the bin numbers. And this is just sort of piler speak for give me a new column called that bin. And we've wrapped this with this group by, which is some real magic in depiler. This now becomes what's called a grouping variable. So basically we tell depiler, I want you to turn this table into a zillion mini tables by taking all the rows that have the same value for bin, and that's a mini table, okay? And then we're going to start processing this whole ensemble of mini tables, okay? Which is exactly what we want to do. Okay, so then we keep processing with depiler, and the next step is to summarize. And summarize is the ante of group by. It takes a whole ensemble of mini tables, and for each mini table computes some way of collapsing the rows so that we can go back to a regular table where the entries are the summarizations of the mini tables, okay? So all of this crap here is to tell it how we want to construct the summary of values from the multiple rows of the mini tables. So for the replicate collapsed mesendoderm percent methylation, I want you to get that from the mini table by taking the mean, the average, of what is also called the ME% column in the mini table. And I want you to ignore the NAs when you do that mean. Right? So that is basically the min in whatever the current bin is that corresponds to the current mini table. And we kind of summarize that column, rename the column the same thing, but now it has, instead of a whole ensemble of values, it's just got the single mean value with the NAs. So we do that for all the columns, basically, all those numeric columns, the coverages, the percents, we want them all summarized that way. But the percents get summarized with a mean, and the coverages get summarized by taking the total, right? So the coverages we collapse by taking sums of the mini table entries and not means, right? If we want the total coverage now, that's what makes sense in the bin. We could do mean coverage too. I mean, that would be okay. It would just change the interpretation. You know, maybe total coverage is more interesting for the bins, right? It's kind of everything's on absolute scale. Anyway, great. We're almost done. It's just that to plot this thing, we don't really want to make plots with the x-axis being bin numbers, right? It's not too crazy because our bin size is in decimal is a power of 10, and so it's easily convertible by I to whatever units you want. You know, if you want megabases, it's add or delete some zeros or move the decimal point or something. It would be workable, but to make a publication, we want megabase pairs or something, not bin numbers. So we introduce a new column, megabase pairs in kind of the center of the bin. We'll let that center actually be a little bit weighted in the sense that if all the data comes from the left side of the bin, we'll have it near the middle of the left side of the bin. And so we'll let the data coordinates sort of pull us towards one end or the other of the bin. So we can actually just do that by summarizing in the mini table. We still got that coordinate column, which is the numerical coordinates of the actual cytosines that are contributing data to this mini batch. And we can just take the mean of those coordinates, right? That's a representation of sort of the logical semi-weighted center of this thing. You know, another way you could weight it is you could actually give the weight higher to the guys that have more coverage or something. It'd be an even more weighted version, but maybe that's not a good idea. Anyway, that's why I call it semi meaning semi-weighted center. It's just based, each site gets to contribute equal weight. It contributes its coordinate with equal weight to this mean. So anyway, that'll give us a coordinate somewhere between the left and right edge of the bin that represents where this data is contributing in this bin. And then to get megabase pairs, we will just divide by 1,000,000. Okay. So, and that is basically the plot coordinate that makes sense for this, this bin. All right, so that is some deep-Euler magic, okay? That would be a nasty business to do in BSeq. And here is what that guy looks like. It's not a data table anymore, now it's a tibble. That is the kind of silly, lighthearted capriciousness that is the tidyverse. It's not a table anymore, it's a tibble. Okay. It is a 6x20 tibble. Okay. Tibbles don't have row labels anymore. They decided that it makes more sense just to keep any kind of label you want as another column, right? It's just, why have this sort of magic row labels, which then you have to have extra syntax for to deal with row. Just the rows just get numbered 1, 2, 3, 4, 5, no matter what. And if you want real labels there, you just make it another data column, so it's the same as every other data column. It's not a bad decision. So we've got the bin column, which we kept, but we're not really going to use anymore. We've got the megabase pair semi-center, which is where we're going to plot as our x-axis. And then we've got these summarized. These are means for the percents, means over the values that contributed in the bin for the percents. And we've also got the coverages, which are the sums of the guys contributing to the bin. So of course, now you see coverages like 4,000 and something like this, because now we've collapsed many CGs together to get this stuff. And including on both strands, right? And oh, what was I going to say? Anyway, it's ready to plot now, basically, right? So we've got it doesn't even show us all the columns, but at least gives us the names, right? You know, this is the way kind of what head does for tibbles. They stop you so you don't just kind of get endless screens full of stuff if you have a really wide tibble. So we are ready to plot. Okay, that was the hard work. So we will make another little mini function here so that the script actually doesn't have to have hard-coded, because I wanted to put the sizes of the bins in the x-axis label, and to not hard-code that into the script, here's a little bit of R code that just constructs the X label. This is a good thing to do so that you know your labels are consistent with what the actual computation was when you start twiddling parameters. You try to have only one place in the script where that parameter is defined, so that everything else automatically updates so that you don't have inconsistencies, you know. So we use paste 0 to construct a string, mega base pairs, bins of, take base pairs per bin, divide by 1,000, that gives us KB, round it, we don't really need decimals and stuff. It won't be super precise, but you're not gonna make a bin size that's not a multiple of KB. Right, I mean, whatever. It's suggestive, not exact. We round it to an integer just to make sure it's an integer, convert it to a string so that we can paste 0 it up, and then KBP done, whatever. So this is our title, mega base pairs, bins of 100 KB, right? And that if we update, in fact, you know, remember I did change this code. It was originally in the slide 10 KB. Now it's 100 KB, but the label is going to fix itself because we do this construction of the label programmatically. Okay, so now the little here's the little, I don't know why I have the little par 3x3 up there. It should logically kind of belong more down there. It's a better grouping. It's a 3x3 grid of stuff. We're going to go through all of our guys again. We're going to pull out now the summarized data. The x-axis is going to be common to all these, so we don't need to give it the x-axis because they're all the MBP semi-center here. So it's the x-axis, so we can just put that in the do. It's a common code. It doesn't have to be in each of these guys. So it takes the y values basically in sync with the x values, which we know, and the title. So there is our x's, there is our y's, there's the x label, there's the y label, there's the title. PCH20 changes the little dots to be little tiny solid dots. And col is the color, which we construct as a red, green, blue triple. That's because I wanted opacity. So this is the red 0 to 1, green 0 to 1, blue 0 to 1. That is dark green, no red, no blue, half green. The reason for doing it that way than not saying dark green was because this is the opacity, and I wanted quarter opacity. And if I take this and make this a little bigger, come on, you can do it. Now you're going to grind on that. I know, I'm not too happy with that. That's a hard plot for you to make. Maybe okay, I'm going to delete that one because I'm just thinking hard about that one. But I'm going to generate the 100 KB guy that takes less work for the machine. And, paste all this. There we go. And I don't want it square. I kind of like it more wide to fit the screen. There we go. Just copy that. I'm going to paste it into the right one so you can see it. This is big as you can get. Yeah, you can see a little bit. The effect is probably actually better with the 10 KB plots where the transparency helps. But there you go. You see these in papers all the time. Okay, how about PCA? We can do some PCA. And we'll do the PCA at the replicate level, right? To see whether the replicates cluster, right? So, and we'll restrict to places where the coverage in each particular guy is 5 to 50. Okay? So there are 6 guys to do. And we'll actually, the PCA is happiest if all the data, the matrix data, the data matrix is absolutely complete, right? No missing values whatsoever. So we are going to pull out the places where each of the guys is between 5 to 50 and then intersect all of those. So they all have to be 5 to 50, right? We're not letting it adapt to each one. We want no missing data. We want actually everybody to be between 5 to 50. So here's the ME1 coverage, 5 to 50. And we also want ME2 coverage to be 5 to 50 and blah, blah, blah. And they are, all 6 of them 5 to 50. That is now a subset of sites. And so we now, that is our row subset. And then actually, we don't want all the columns either, right? We don't want a PCA to treat all those columns as different things to PCA. So we only want 6 columns to be done. And that is the ME1%, ME2%, MS1%, MS2%, ME1%, and ME2%. So that is the percents at the site level for for the 6 samples. So I'll call that whole CG cut. Right? This is from the whole genome, but we have cut to this subset of common sites. We can find how many sites that is. So whole CG had, no, it's not size, it's dim. Whole CG is 21,671 rows, and this whatever we just made, whole CG cut, is what it's called. That is 14,911 rows. So cut down, but still we got 15K. Right, and if we look at it with head, you'll see that we've only got those 6 columns left, right? And if you look at these row numbers, it's a sparser set. If you look back, so that is the data we want to PCA. Although the PR comp function in base R doesn't like the data this way. It wants the data transposed from this. So it wants the rows to be the vectors you're PCAing, and the sites to be on the columns, which is backwards from the way we've got it right now. So we got to flip things around. Now, for stupid reasons, it's transposing data tables is messy in R, and all kinds of things can go wrong. And so we're going to actually use, it may or may not happen in this case, but it's better not to think about it. So whole CG cut is a data frame. Transposing data frames is fraught with all kinds of difficulties in base R. So there's this library data.table, which contains a transpose command, which is a little safer for transposing data. So this transpose comes from this library we just loaded here. And so that will flip it around. It also loses the row and column names, so we'll just put those back. So the column names of the transpose guy are the row names of the original, and the row names of the new guy are the column names of the original guy. And so that will flip things around. And it's now 6 by 14,911, which is the right direction, PR comp 1. Okay, and then to get the nice labels in this plot, we got to build a little key too, which is going to be a little an extra. Why don't you like that? I expect to input in something like comma or something. Maybe I was just sloppy with the selection. Maybe there's a. Yeah, I've got something. I've got like a shift return here. Yeah. Let's see. Okay, maybe I've got a shift space. I'll tell you that story if that ends up being a problem. Oh gosh. No. I don't know what you've done. You've gone to a slide that's not even supposed to be there. We've lost our navigator. Gosh, Keynote, F5 replaced that with Violent. Nope, that's too far. Not there yet. Almost there. There we go. All right, I'm just going to try line 1 and line 2. There's something funny going on there. Okay. All right, got a plus sign for line continuation. That's good. It's not too unhappy yet. All right, that works. Okay. So key is a data frame that has row names, which are, we can make these nicer than this, but it's basically the column names of it's the samples that we want PCA to PCA up, right? So it is the mesendoderm replicate 1% CG methylation, per site CG percent methylation. Same thing for the other riots, right? So there's 6 of them total, 6 samples. Then there's a column called tissue, and that tells the grouping, right? Because we want to color the dots by the grouping. And so I just took the first 2 characters, which is the grouping, right? So the mesendoderm is the MEs, these are the MSs, and these are the NEs, and it's convenient the way we did the names. We can just take the first 2 characters, right? Which is what this business does here. So the row names, TR, that's those, and then the subster 1 to 2 takes characters 1 to 2 out of those, and then we want that in the tissue column, and that's how you build up data frames like that. You just say column name equals data. Right? It's data frames. Row dot names is when you assign if you want the row names. Okay, so we got this nice key, which will be used to, this tells the auto plot that when you need various decoration actions, they should come from this table. And then the color of the dots comes from the, you should group it by the tissue column. Okay, so this ggfortify library, which is based on ggplot2, is just nice because it understands, it knows a bunch of our objects directly, and it knows how to make nice plots just from the class of the object it's given. It's just magical, which is good for exploratory data analysis when you want a kind of a nice looking plot without doing a lot of work. So I'm going to load ggfortify. That gives me autoplot. Here is the data ready to go. PR comp, that's the PCA analysis of base R. Autoplot will figure out nice, autoplot knows how to make nice labels and all kinds of stuff for that. This is our metadata information so that we can group, make color, it can make a color key and color the dots based on the tissue column. The label says I want the actual names to be written next to this, the dots. The label size 3 makes the labels rather small because they're quite close together, some of these dots, and bigger labels would overlap. And the real magic is this label.repel equals true, which makes it so the dots labels are not going to overlap. It sort of does a little simulation, and it has each of them start sending out repelling energy to each of the others. And then if they're too close, they'll start pushing each other away, and eventually it reaches a steady state where the labels hopefully aren't overlapped. And so that is how we get a pretty nice looking plot without being too careful about, you know, having to do much work to get those labels to look reasonable. That's the real gold there, is the label repel is true. So that is our PCA plot. Piecing component 1 is mesenchyme versus the other two. PC component 2 is the two replicates of that against each other. So that is a more variable guy, right? Okay. The two other guys, the replicates pair better than they are to each other, which is great. So I mean, this isn't that crazy, right? I mean, we should maybe start wondering about why this dimension is so big, but otherwise, not too bad, right? There's all kinds of parameters that share here and so on, but basically, you know, that is more or less how PCAs go. Okay, so that is the end of what I wanted to show in R. I will take any questions, we'll have a short break, and then the last bit of today will be the differentially methylated region detection and just a tiny bit of trying to throw that into a functional annotator. So anybody got any questions? How are you doing? I mean, you're following along, you got the plots to work. You guys know R pretty well, or R is still pretty new to you? I think R is a requirement of the workshop nominally. Anybody want to contribute any chat stuff? Or? Good. Yeah, I'm trying to give you a whole bunch of, you know, examples to kind of use that you can build from. So you can tweak this code as needed, but hopefully it gives you exposure to some R concepts and libraries and functions and so on for getting stuff done related to this kind of data. All right, so we got 45 minutes left, and that'll be enough to finish off the demo. I mean, you know, we could go on forever. We're just going to cut it at some point. The goal is really to give you kind of a really solid basis on the primary analysis, the mechanics of it, what each of the files means inside, and so on, and how to manage them. And because without that base, it's kind of hard to do the downstream in a good way. The downstream is more specific to what kind of project you're doing, and it's easier, I think, to pick up. You can't do it unless you have the base analysis in good shape. Okay, so let's try some differentially methylated region identification better than what just browsing around randomly in IGVY was doing for us. So for this, we will use Methylation, which is a C program out of Germany. And it's pretty easy to use. It has some parameters which are good to experiment with. And I'll show you how sort of interactively we do that. Unfortunately, Hoffman2 doesn't have any version of it installed by base in the module system, but it's fairly easy to get installed. So, okay, we're done with R now, so R is going to go away. And we're going to go back to our shell window. And that is my local shell, which I'm also done with. And then this is my Hoffman shell, which I'm still in the stage 7 subdirectory, which I'm going to back out one of them. So now I'm at the day 3 directory on Hoffman. Okay. And somebody tell me if this window is still visible. Do you see my Hoffman terminal window? That's still screen shared, I hope. Yeah, okay, great. Okay, so as I say, it's not installed. We got to install it. It's a C program, so it's different than the other ones that we've done today. Okay, so here is the URL to grab the current version. I mean, here's the general web page, which you can browse on, whenever you have time. You just basically click download in there, or get the URL of the current version or whatever. And here's this little trick for capturing the output of wget into just a little documentation file. So I'm going to paste that in, and we're going to get the source code for this thing. Okay, and there is a tar gz file, which is basically a zip file, but it's the Unix-y like zip file. It's a gzipped tar archive. Tar comes from probably before some of you may have been born. Tar is a very, very old programming file format. But it originally stands for tape archive. When magnetic tape, in the movies, when you see the big, the movies from the 50s and the 60s, and you see the mainframes with the big tape spinning. Tar was the program that was backing up your data, basically, to tape. Still around today. Basically, it takes directory trees and turns them into streams of bytes, which we can then store in files or do whatever we want, send them over the network and so on. Anyway, it's basically one of the ancestors. It's a functional ancestor of zip. Zip just took off on the PC world. Okay, so we got the tar. We got to open up the tar. Now, like with all archives, tars and zips and so on, you generally shouldn't expand them until you have some idea what's inside them, because they can have relative paths. They go to parent directories. They can dump stuff right in the current working directory, which sometimes if you don't do it and you don't realize it, and it's a large tar or something, it makes an absolute mess. Now you've got files interleaved with your own, and it's very nasty to clean up and so on. So it's cheap, so just use tar tvzf. The T means give me a table of contents. The V means show me every file as you go through it. The Z is because it's gzipped, we need to g unzip it, and the F means the next argument is the name of the tar file. So this just shows me what's in there without expanding. And you can see that all the paths are to a subdirectory that it's going to make. And it's not. This is a hygienic tar. It's a tar archive. It's not going to dump anything on us where we are at a higher level or do anything. You can even have absolute paths in tars. So this is fine. We have nothing to worry about there. So we're just going to expand it, and that is xvf. X for extract, V to show me each line is progress information basically, and the F means the next command line option is the file. So there it is, and now we have this Methylation B02-8 directory, which is where this source code has expanded to. Now we can look in there, and we see, what do we got? We got a make file, we got a README, we got some kind of PDF file which we could read, but we're probably not going to, because yeah, we're going to just try to do this like everybody else does, which is to not read documentation. And then we got stuff that looks like could be executables already. So maybe they've given us executables already. So actually we could use the file kind of heuristic identified by content detector to try to tell us what this stuff is. Okay, license is a plain text file. Make file is a make commands text in plain text. README is plain text. Manual.pdf is a PDF document. Input.pl is a Perl script. Linux 32 and 64 actually are Linux native binaries, 32-bit and 64-bit. The .r is an R script, and the PL is another Perl script, and the source is directory. So we could try to run these guys. They have an XBit. I mean, we just downloaded a binary from a random place on the internet. whatever. But unfortunately, it doesn't work with Hoffman2, and it's got a glibc incompatibility. And so we're going to have to compile it from the source code, which is in the source vector. Here's all the .c and .h files. This is the C programming language. You know, you've never seen C before. But, this is the language that Unix was originally written in, basically, and it really took off. And basically, every real big program gets written in C or C++. Python itself is written in C. Many Python modules install C bits internally that they can run faster, and the Python is really just more of a glue language that just calls out these little C bits and so on, and gives you a nice syntax and nice data structures. So we're going to have to compile it, which means the makefile. I mean, there's this README. We can read the README. And so it says, okay, you know, all installation, just type make, try to precompile. That doesn't work for us. Run Methylation, just call the program, blah, blah, blah. Okay. What about the makefile? Makefiles are another thing you shouldn't just run kind of randomly. They are basically shell scripts. And they could contain RM, RFs that delete your whole file. Who knows what? I mean, the question is, do they have parameters? What directories are they going to try to stuff in and whatever? And this one is actually a very innocuous one. It's amazingly simple. Usually these things are nasty. There's no commands here that look remotely threatening. No hard-coded directories or anything. It's all relative. Nothing to worry about there. That is about as easy as it gets. We just type make. It's calling the new C compiler to create the binaries, compiling each .c file to a .o file, and then a final linking step where all the .o is turned into the executables, and it made this executable Methylation. So if I ls-lat, the T sorts the listing by time, giving the newest files on top. Certainly these are 2018, these were the ones that expanded from the tar. The source has probably been hit because we created O files in there and so on. But the real interesting things at the top level here, we've created this new file, Methylation, and what is that? It is a Linux executable. If we ask the system for the shared libraries in it, can you find all the shared libraries that it needs? The answer is yes. We can try running this thing, and there we go. We get some help. It looks like we're in business. Okay, so we've got Methylation installed. I will back out one level. When we need to run it from this level, we're going to have to give a relative path. So Methylation, right? So that's how we will invoke it from this place. All right, so now we're back to our usual pattern of dealing with a new tool or a new module of the tool. So, okay, we did that. Here's the help. What kind of command line options look relevant for this thing, for getting it to work? So the input file, which it doesn't mention what columns it needs, but I'll tell you right now, it basically takes CGMap files as inputs. The only difference from the CGMap files that we have versus what it wants is it needs that first column, which is chromosome name, :coordinate, to be separated into two columns, which is chromosome name in the first column and coordinate in the second column. That's the only difference. We got to take care of that. That's the only thing that really matters for getting this thing to read our files. What does Methylation do? So it tries to call differentially methylated regions. How does it do? Okay, so it's basically, in a way you can think of it, doesn't work this right inside, but you basically think of it as it considers every possible starting and stopping point for an interval on the genome, right? And then it looks at, it's an A versus B comparison, right? It does two conditions at a time, condition A versus condition B. It's a pairwise comparator only. And A will have one or more replicates in it, one or more samples, and B will have one or more replicates in it, samples. And you can think of it as, I mean, you know, you have to read the paper or whatever to see what kind of test it's really doing. But the conceptual situation is essentially for each of these intervals, once you decide on the intervals, so it says this piece of genome here, then you've got the methylation values for all the cytosine sites in this interval in sample 1, sample 2, sample 3, sample 4, all the samples in group A, and sample 5, sample 6, sample 7, sample 8, or whatever, all the samples in group B. So you've got a bunch of observations here and a bunch of observations here. And now you need to do something like kind of a two-sample t-test, or you're doing something like that, you know, wherever you got to tell me whether the mean in this group is different from the mean in this group on this interval or something. That's basically the kind of thing that it's trying to do. And then, you know, if the literal region on the genome that actually is differential right? Suppose it's very, very clean, right? Suppose it's completely CG methylated out here. There's a very sharp boundary where some of the samples just stay completely CG methylated across there, and the other group, the samples just go to 0%. It's completely, an easy one, right? So it's in IGV, it's just solid red, solid blue, solid red, and solid red all the way across, right? In group A and group B, right? That's the easiest possible situation you guys for, right? So if you have an interval in the middle of that blue region or something, it looks pretty good, right? Because it's like all blue versus all red. But if you made the interval a little bigger, it's even better, because then you've got all blue and all red with more data, right? So TF will like that even more, right? That's even more observations. It's stronger, more power, and the difference is still huge. So it's bigger. So essentially what it does is that once it gets its foothold on a region that looks different, it optimizes the edges to try to get the best edges for where the region gives you the most difference in the test. And so it basically walks along the genome at a certain tiling, and when it kind of finds a candidate region, it then optimizes the partitioning and so on, and it just kind of walks along and optimizes as it goes. And because DMRs are not usually allowed to be very big, we don't have to look a megabase away to sort of optimize what's going on in this region here. It's just not effective. And so it can basically do a very local picture walking along and optimizing businesses, and it doesn't really need to load the whole genome into memory or anything. It can work very locally, and so on. And so that's what Methylation is going to do. And the output is going to be a list of, the regions that pass the statistical test, the optimized regions that pass the statistical test. So it's going to be chromosome start coordinate, end coordinate, some kind of p-values, and maybe an FDR adjusted Q value, false discovery rate adjusted. We're going to have the actual mean difference between the groups, right? Strength of effect, right? Statistical significance is one thing. Statistical significance is a combination of, how much data and how big the effect is, right? Whereas as biology goes, we're going to want to know the size of the effect, right? Because we also like big effects, right? And then well, that would be about it, I guess, right? That's the key information. So, and that indeed is going to be what's basically comes out of this thing, in plain text, type separated text. That's kind of the information that's going to be there. Okay, so what do the parameters look like then? We have the maximum distance that we have, 300 base pairs. This looks like some kind of thing that's related to internal workings and memory. We'll just leave that alone. The minimum number of CPGs to be in a region, default 10. Maybe we'll try 5. Maybe we'll raise this to 1,000. Minimum mean methylation difference. So this is the minute, and it looks like it's expressed as a fraction, because 0.1 sounds like 10%. It sounds like it's a fraction. It's not 0.1%. That would be good. It's probably a fraction, and that means 10% then. So maybe we'll increase that a bit, because 10% is a pretty small difference, especially with the kind of coverage we got. So maybe we'll make that 25% or something. So that is the mean difference between group A and group B. This is how many threads, number of CPU cores to use. Here is, it can operate in a mode where if you've already got DMRs declared through other studies you've done or whatever, you can score a new guy against the known DMRs rather than this de novo discovery of optimizing the boundaries and finding the DMRs from scratch. So this mode thing is whether you're doing that. 1 is de novo, that's what we'll do. 2 is predefined regions, and 3 is another thing, okay? And MTC is the multiple testing correction. Okay, MTC, multiple testing correction. 1 is Bonferroni, which is basically just divide by the number, p-value divided by the number of tests that you did, right? The p-value should be penalized by the number of tests you did, right? Just by multiplying or dividing, depending on the point of view. And 2 is probably these days fairly commonly used, very widely used, Benjamini-Hochberg, or Hochberg, I don't really know how it's supposed to be pronounced, false discovery correction, which turns p-values into q-values by mushing them around a little bit. But the Benjamini, the BHFDR stuff never changes the relative order of things. So if you're interested in rank order, it doesn't really do anything, right? It just changes maybe where your threshold is, but rank order does not change under the BHFDR transformation. And then we have to tell it which samples are in group A and which samples are in group B. And the way it does that is in a way it's not flexible, in a way it's nice, and it's a way that we've set up to work very well with the way we got things right now. What you do is you give so the input, right, is going to be like one of these aggregated, we made this aggregate matrix thing we ran from BS-Bolt to get our data. It's the data we imported into R, right? It basically wants that kind of data as the input, right? And it doesn't take the count. One of the reasons why we made both the count version, which was useful in R, especially when we looked at coverage, and the fraction methylation version, is because the most appropriate file to feed to this thing is the fraction methylation, not the coverage. We don't want differentially covered regions. That would be noise bueno. That would be, we want differentially methylated regions. So the number that we need it to use to decide when it's, comparing numbers between the two groups, the number needs to be compared is fraction methylation. So that is why we have that flavor of file that we made. We have the aggregated matrix fraction methylation flavor. That is essentially the file we want to feed to Methylene modulo the splitting of that first column into two columns. So that's easy to do. So how did you know which is in group A and which is in group B, which samples in group A and group B? Well, that aggregated file, remember, has a header line that names each column with the sample names. And for us, there are things like, neural P underscore ref1, stuff like that. Right? What you do in Methylene is give a string prefix of those column titles. And the columns that match the prefix are the ones that belong to the groups. And since we put the tissues, tissue underscore as the beginnings of the column names, the sample names, it's very easy for us to do in Methylene. Right? So we will just, like, one group A setting could be neural P underscore, right? And then all the columns, the fraction meth columns whose names begin with neural P underscore, which is exactly the neural progenitor condition, the two samples in the neural progenitor tissue. Those are the ones that become group A. Anyway, so that's how it works. Better, of course, make sure that your sample names don't have, like, one reason to put that underscore there is to make sure that none of them can be a prefix of each other, right? If they all end in a single underscore and your pattern includes an underscore at the end, it means that you're kind of anchored the pattern. So don't name your sample names with things where, like, one could be a prefix of another, right? If one tissue's name could be a prefix of the other, you're going to have problems in Methylene because you can't isolate the groups right and so on. But usually it's not a problem. But you can imagine, you can contrive it as cases where it would be a problem. The bad argument is if you're in this predefined region mode where that's not relevant to us. You can put thresholds in how many samples contributing values to group A and B. We'll just let that float. No constraint there. There's a random number of business in there. You can control the seeding and the value filter. We'd have to go look at the paper and see the algorithm for this thing and so on. And so this is some parameter that controls how it works in the partitioning and so on. And we're just going to leave it for default. So the ones we really, maybe we raise this a little bit, maybe we lower this a little bit. We're going to increase this a little bit. Threads. Well, in my current session, threads should be 2 because that's how many my environment was asked for when I started my interactive session. This is going to be 1 for de novo mode, 2 for Benjamini-Hochberg FDR correction. This is our groups. This will be our sample name prefixes to define group A and group B. That's not relevant. We're going to leave those alone. We're going to leave this alone. This basically we're going to leave as a default. And so that is our plan for running this command line. Okay, so that's all good. So that's basically what we talked about. Oh, and it requires the input file to be sorted by chromosomes and genomic positions. Well, the aggregate matrix BS-Bolt did that. It guaranteed that. So we don't have to worry about that. All right, where are we now? Okay, blah, blah, blah. So here's our usual thing. I'm planning out the template that we're going to fill in with parameters to see how this goes. Is the command line we're going to run is going to look something like this. We've got this issue where we got to deal with the first column of our ag_fur myth files needing to be split into 2 columns. So we got to figure that out. As it turns out, like I said, Methylene reads its input basically in streaming mode. It just basically walks along the chromosome left to right. That's the reason why it requires the file to be sorted by coordinate and so on. And so it doesn't really need to seek around the file or whatever. It just kind of reads the file straight through. It doesn't back up or jump forward. So it reads it in what's called streaming mode, no seeking. And that means we can actually generate this column 1 to 2 column split on the fly. We could make a whole set of parallel files and just have them sit on disk with the 2 column version. But We can just generate the split on the fly. So this is where you would normally name the path name of the input file, and instead we can use the bash shell less than parens construct, which means set up a pipe, run this command line, take the output of it, as you know, we're kind of, this is a running thing, and it's almost as if there was a little pipe symbol here at the end, and the output of the pipe is turned into sort of a virtual file that then is given as the command line argument here, and which Methylation will read from. So it's essentially Methylation is reading from the output generated by this mini pipeline. And what is this mini pipeline? We're using sed, which is another thing like awk. It is a text stream processor that takes lines of text on input and can transform them through configurable little ways, like little mini programs that you can fit into command line arguments for simple transformations. And it produces output on standard, standard in, standard out, little transformations on the way. It's kind of exactly what we need to do. The -E introduces a command for sed, and it just kind of, for each line, it just runs through all the commands in order over and over again. The first command is a regular expression substitution that changes colons to tabs. And we need that done in every, we need that done not just the first colon on a line, but all of them. Why am I thinking that we don't really need that G there? But, well, let's go take a peek at what we're going to give as an input file here. So that is going to be in our aggregation output here. It's the ag-for-meth versions. So that's, here's like here's the CG, CPG context, ag fraction meth. And each line only has a single colon. So I'm now confused why I asked for the G there, which means replace all colons. It won't hurt, but I don't think you should need that G. Okay, the other thing we should do is we should update the header line. And given this file is of very uniform structure and our chromosome is just 21 and absolutely no risk of having the word site show up in the chromosome name, we can do this with more or less safety, but for us, certainly if we just search for the word site and replace that with the word chromosome tab site, that will fix the header line to represent the fact that we now have 2 columns. I also haven't quite played with Methylation to the extent to know exactly what it can tolerate on the input. Maybe we don't even have to do that. Maybe it just ignores the header line. Maybe, anyway, we could find some experimentation required here to find out exactly how careful one would have to be here. Certainly, the example as written here, this will be fine for our situation. So let's preview what this will do and see if this sed transformation is actually going to achieve the required transformation. So, okay, there's the file names. There's the sed command, and let's feed it one of the actual file we can feed it, which is the CG ag-for-meth here. And we'll type that to less and see what comes out. Oh, that looks great, perfect, right? So here is the original column header for this thing turned into per 2 columns with titles per site, and then the colon is gone, replaced by a tab, and so now we've got the things broken. Right? So that's exactly what we need. And we can just generate that on the fly, and Methylation will be happy. Okay. Now let's actually do this interactively in our session. But rather than actually writing a bash shell script and put it in a file, I'll show you bash is really a complete scripting language by itself. It actually has functions, right? So you can actually write functions in bash. Example, right? And here we go. So let's take we'll call this plus. It takes 2 command line arguments, which we'll rename A and B, and then we can compute the sum of A and B as a number. This is this paren double parens is a way to do arithmetic, integer arithmetic in bash, and then we'll just produce that output. We could compactify this even though it doesn't have to be this spaced out. But anyway. And then we don't really want to change the environment variables, the shell variables in the caller, and so we could make these local variables in bash so that all these variables just leave the calling environment alone, and we are just playing with our little local environment inside this function. Okay, and now we can do +39 and outcomes 12. Okay, so anyway, we can do functions in bash, and that could be helpful. So this is going to be First of all, we're going to write a little function. I'll show you what it does, and then we'll see how it works. So there is the function, and all you do is give it the context you're interested in. Well, not with the underscore. So the context, as far as we spell them for the point of view of these files, is like this, right? And its job is to just construct the path to these things, right? So this is basically the path to the CG version of the file. Oh, because it's assuming it's assuming your paths are from the day 3 point, not from the subdirectory. So yeah, so you're interested in where the CG context file is. So it'll tell you it's this, and then if you look at that, that's the file. Okay, so that's just basically a file name generator that lets us just give the part that we're really interested in varying, and then have it give us the full path to the relevant thing. Yeah, so I showed that example. And here is, we're going to make a little Methylation function, because Methylation has all these parameters like, oh, you know, the min CPGs and the diff level and the ag map and the max dist and that valley parameter. And we don't know yet what parameters to use. And like, we could generate, you know, files for each one, download the file, sort the file, pull out some regions, go look them up in IGV. But while we're in sort of this exploratory mode, it'd be nicer to sort of interact with things faster and be more rapid. And we'll fixate it into files and so on at the end when we kind of know what to do. But at the moment, we kind of just want it to start generating some intervals. We'll check them out in IGV. And so we just want to play around a bit. We don't want to have all this junk on it. So this little function will kind of allow us to run rapidly through Methylation permutations. So we're just going to make all the parameters that can vary, be command line parameters to this thing, and we'll just have them positionally encoded in a particular order. So min CPG first, max is second, min def level third, valley fourth, the A group sample name prefix, and then the B group sample name prefix, and then the file name for which ag format you want to put as input, which is basically the context. And then it turns out Methylation does not output a header column for its output, which is fine. I mean, once you know Methylation, you know what it is. But just to help us out, it's just at least a memory jog while we look at this on the screen and so on. Here is the column key for the Methylation output file. It's going to have a chromosome column. So this echo is just going to give it the names with spaces between them, but they'd rather be tabs because the Methylation format is tab separated. So it's too hard to type the tabs here. You know, we'd have to do all this crazy stuff, right? You know, we'd have to do this kind of thing to get the tabs in there. But it's just much easier to just put spaces there and then type it to TR and change the spaces to tabs all at once. Okay, so first column, chromosome. It's like I said, it's producing intervals, right? So chromosome, from coordinate, to coordinate, FDR just Q value, the observed difference in methylation fraction between the two groups, the number of sites in the interval, two p-values actually, a Mann-Whitney test p-value and a Komolgorov-Smirnov test p-value, and then the actual mean value of the methylation in group A and group B, and the difference of these two means should be the same thing as this column over here. So that is the stuff that comes out of Methylation. And now we're going to invoke Methylation. That is the path relative to the day 3 place we plan on running this. That is the de novo discovery mode. That is FDR, BHFDR correction. That we're not going to change. And then we take all these parameters that we arranged to pass to the script and we now just feed them in. Okay, blah, 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 feed all these in. Okay, and then here's our little sed trick that does the 1 to 2 column split on the fly. Okay, and then we package that up in a little shell function. So, moving on, okay. And we're about ready to start experimenting. So let me not even run this first one. That's more related to when we get to the point of fixating things in files. This is an example of how we're going to run this thing for a little while. First is we're going to do only CG context. So all that structure I made to really parameterize it for different contexts, because maybe you are in a situation where you have different contexts. We're not going to really vary. So let's just run that path name constructor at once, and now we have a shell variable that has the 201,671 sites that we've got right now. These are the sites that have min coverage for in each sample and have at least 4 samples with present data. That was what we decided to construct the aggregated guide. Okay, so that's just a shell variable that gives the name of that thing. We're not going to change that. And now let's start running Methylation, right? Okay, so I'll show you just without this extra stuff, and then I'll show you why I have that extra stuff. So, for playing around rapidly to avoid, just to keep things compact and so on, we'll just keep the parameters by position, right? We sort of decided somewhat arbitrarily that this was the order they were going to be in. And so we can just put things in here. There's a Drosophila that's been bothering me. It's going to be too hard to get. Yeah, so copy this. Okay, so the 5 is the number of CPGs minimum required to be in the region. This is the max distance there, the max size. This is 25% minimum difference between the groups for the percent methylation, and this is this as yet somewhat mysterious value parameter. Here's the 2 big guys. This is we're going to compare mesendoderm versus neural progenitors, and this is the the aggregated file we're putting input, which is fixed to the CG version. Okay, so we run that, and well, Methylation gives you a lot of progress information, and then it gives you the intervals, and the intervals scroll off our screen, and as it turns out, they're not sorted by Q value either. So to make this interactive a little more convenient for rapid experimentation, let's get rid of that progress information, which is going to standard error. So we're going to redirect standard error to a dev null, which is the big circular file, bit bucket in the sky. So standard error just got thrown away. Now we just get the output, which consists of the header line we added on there as our memory jogger and the actual data. But like I say, for kind of experimentation by hand right now, we don't really need the full list. We just need kind of the top of the list. It's not sorted by Q value, which is column number 4. So before we start taking the top of it and cutting it down, we better sort by column 4. So sort K44 sorts on column 4, but we have to tell sort what mechanism to use. Should it compare it by dictionary strings, or what, is it integers, ascending order? No, it's sort of a free format C style float. These are all okay. None of these show scientific notation actually, but you can kind of see here that sometimes the floats use scientific notation, E minus 05, 10 to the minus 05. That could happen in Q value too, although it's less likely because the Q values tend to get adjusted to fairly pessimistic things here. So to make sort understand the possibility for things like this in the float, you have to use the G, which is general number. So that is ascending sort as a C-style float on column 4. I could run that again now. Now you will see that we are sorted by the Q value. So good ones at the top. There's also some 0.02, that's okay. And we're just doing chromosome 21 here, right? We've come down to a really small fraction of the genome. To get anything out at all, we should be happy, I guess. And then let's just, for experimentation, cut it down to the first 20 lines. So that's basically the top 20 by FDR-corrected p-value. And that gives us something we can kind of look at a screen full at a time and maybe start kicking over to IGV and start checking these guys out. So somewhere I lost all my Zoom controls. So, oh, they're up here now. Okay. All right, so I gotta bring IGV into the mix now. So share screen. We need Keynote, that terminal, and IGV. Wait a second. I don't know if Ela has done something or not. I can no longer share my screen. Okay, so here is IGV. Let us put in one of these regions. Okay, so I actually happen to know, I think it's the second region that I like more right now. So I will show you the second region. This has got a Q value of 0.04. Maybe if your 0.05 is what you like to use, that's right on the boundary. We haven't played with these parameters to optimize it. So maybe we make that better or not. But actually, another thing to do is just look kind of at the raw p-values, right? I mean, anyway, so that the Mann-Whitney test is around 10 to the minus 6. The KS test is around 10 to the minus 6 too. Plausible. It's not too bad. Let's actually look at an IGV though to see just kind of what we actually got, right? So I'm going to copy the chromosome coordinate business, paste it in here. Now I think IGV doesn't like that, so I think you have to put a dash there, and we got to put a colon. And we are there. It's probably where I already was, so nothing changed. And now that's the 535 base window that Methylation decided, but let's see it in context. So let's zoom out a couple steps, right? So now I'm at 5 kb, right? So now as I scroll down, remember I still got this IGV that's got this issue where it seems to have enormous amounts of white space vertically between my different tracks, which I haven't figured out yet. But it's okay. We know that the interval of interest for Methylation is dead center, and we're just going to scroll up and down and see what we got, right? So, and we'll talk about what we're inside some gene annotation or something here. We'll check that out in a second. Let's just look and see whether we've got something that looks good. Okay, so here we're scrolling down. Let's just scroll through it first once, and then we'll go backwards, and we'll see whether we're like, wow, it looks good, right? So here, I think which two samples we compared, but red. IGV is showing 2 strands. I've got it grouped by strand, plus and minus strand, right? And then 2 samples in each tissue. So there's 4 IGV rows kind of per condition. And that's red in the center, and this is blue. Okay, so that does look like a differentially methylated region, right? And that looks like a pretty good one. That's 500 some bases wide, and it looks pretty strong. That looks quite acceptable. We could look at the tissues and figure out who's hypomethylated and who's hypermethylated. That's unfortunately I didn't name my band tracks in a way that makes that easy. But we're also running out of time, so I don't want to take time with that. What gene are we in, right? So if we start hovering over these, what gene are we in? We're in RUNX-X1. Okay, RUNX-X1. All right, let me get Firefox in this. So. Okay, so now I need IGV, I need terminal, I need keynote, I need, why do I not have a window with Firefox? That's within the list of things to share. No, just took it a while. Okay. Firefox, IGV, terminal, keynote, go. Okay, so the Googler. All right, so that was RUNX-X1, DNA. Let's know. All right. DNA methylation of RUNX-X1 regulatory regions correlates with transition from primitive to definitive hematopoietic potential in vitro and in vivo. Well, this is sounding, plausible. This looks like, could be some real biology, which essentially we've discovered, right? I mean, we could have been in a discovery mode here, right? We've got the next region. All right, so we're running out of time. But basically the way this is set up now is that you can rapidly tweak these parameters. Like, okay, suppose I change that one to a 10, right? What happens, right? And if you make your window big enough here, you can actually see the previous run too. So you can sort of see are your regions coming or going. It's like this region here seems to have moved to the top of the list now. The Q values got worse though. Anyway, so you can play around. There's this one. And anyway, you get how it goes. I could change. You just play with them, right? You can play with them. This isn't a rapid kind of experimentation. So anyway, and then one thing people do is that you can check for functional enrichment of these. I mean, obviously you're going to check them out in IGV and so on. You're going to see what genes are near them and so on. If you did this now, I don't think this works for the chromosome 21 analysis because there's just not enough good regions here. You really need to do this for the whole genome. But there is these tools like GREAT is one of them. So GREAT analysis, I think enough to put it in Google. genomic regions of Google had the title there. Google genomic regions enhancement of annotations. I don't know what the T is. Yeah. So here's the page. And what you basically put in is, we would have to tell, it only knows these genomes, right? So we have to tell it we're using GRC-H38 as the reference genome, which is ours. And then you have to give it the coordinates of the intervals that you're of interest. So for us, it has to be in bed format, which basically is the first 3 columns of this thing. So, you know, I could just rerun that again, but give me the first 3 columns. All right. Copy and paste some of these guys here, background regions, blah, blah, blah. And then because these regions are, you know, have to be, we're in the middle of, you know, genes have long transposons and humans on long introns. You have to have some rule for how you sort of convert this intervals into the genes that it's going to really use to search against the functional annotation. So, you know, we could see quite generous. It's like, oh, you can go a megabase up and down to find candidate stuff, you know, whatever. All right, and then you say submit, and then it does an enrichment test and tries to find out whether this is And in great speak, the chromosomes need to have CHR on the front. We can take care of that. We can take care of anything, right? So we'll just do a little awk on each line. We'll print CHR %S followed by the right. That prepends CHR to everything. Okay, now we got something we can calculate. Easy. Okay, GRC-H38. GRC. That data. Blah blah blah. You probably lost this. Yeah, you did it. Okay, here we go. Submit. Thinking. Shouldn't take. That's not many regions. I mean, normally you put in like 100, 200 or something. It's like a, you know, it's a gene set enrichment test kind of thing. Come on, this was like instant yesterday. Oh, there we go. Okay, so it tells you the intervals, how far away they are from transcription start sites, is essentially promoters, right? This interval happens to be, the intervals we gave are kind of far away. Whether there's any enrichment tests, actually we have some molecular geontology molecular function enrichments, okay? Type 1 interferon receptor, interleukin 10 receptor. Anyway, so you know, obviously we should do this on the whole genome instead of just chromosome 21. There's parameters played with this one, but gives you the flavor of it. All right. So hopefully this workshop was helpful to you. If you're taking it for credit, look in your email for the quiz and the homework, and get me the quiz back by Monday end of day, and the homework back by Tuesday end of day. And do the feedback that, you know, any feedback you want to give that I'll give you the link to or something, that would be appreciated. And hopefully this is helpful to you guys in your real research, and we'll catch you at the next workshop I get. So I'm going to stop recording. And